Hey everybody, thank you very much for stumbling upon this 5-hour crash course video for everything you need to know about clinical research, whether it's getting your start in the industry, whether it's to expand your career. This is an accompaniment to the book, The Comprehensive Guide to Clinical Research, as well as the Audible book with the same title. It's not required that you read that book to understand all the information from this video, but it's a great companion. We're able to go into a lot more detail. I tried to keep this video under 5 hours, but the book, it's a solid book, and then the audio book is almost like 2 books, because it's the book itself, but then Chris and I, the authors of the book, go off script. So this video, I hope it's able to help you. I had a 2-hour video in the past. I thought it wasn't enough. We needed more. So this is a 5-hour video about everything you need to know about clinical research, from the site level, the CRO level, the sponsor level, the vendors, the regulations, the career paths, all the fundamentals, source, regulatory. It's all there. If you have any questions, email me, dan@theclinicaltrialsguru.com, or call or text me 949-415-6256. Thank you very much for watching. Hope you get something out of it. Take care. Hey everybody, welcome to YouTube. Welcome to the Clinical Trials Guru. This video is going to be the 2019 version of the Introduction to Clinical Research, and it's actually going to be the video that's the foundation for the book, Comprehensive Guide to Clinical Research, which probably by the time you're watching this is going to be on Kindle, on Amazon, on Audible. We're going to go off script a lot. Me and Chris are writing this. We have a couple other people helping us out doing the research, researching the different regulations like GCP. It's going to be a beginner's guide, and this video, I don't know how long it's going to take, but we're going to go through most of the book and give you a summary of pretty much everything you need to know to get started in clinical research, either as an employee or as a site owner, because there's a lot of people out there that want to become site owners. So let's get right into it. The first thing we got to look at, the first thing most people get confused about with clinical research is they're missing the bird's eye view aspect. So you basically, clinical research is unique in the sense that it's where everything in the biotech and pharmaceutical industry, it's where all the bottlenecks occur, but it's also where all the innovation happens. And it's the same thing. It's clinical research. It's responsible for the bottlenecks. It's responsible for all the innovation that happens in medicine. So this is obviously unfortunate, the bottleneck part, not the innovation part, but it also contributes to the high cost of drugs, devices, healthcare in general. So we're going to get into the overview of clinical research, okay? So when a drug company, all right, so I guess we should start there. Drug companies, biotech companies, medical device companies, diagnostic companies, whatever the case may be, they are called sponsors, and we're going to call them sponsors for this book, and we're going to call them sponsors for this video. So the sponsors are the ones that run the whole thing, okay? They finance studies. These are sponsors like drug companies, okay? There's also other sponsors. There's 2 types of sponsors. There are industry sponsors, which are all these drug companies, and then there are academic institutions or grant recipients, usually from the NIH, the National Institutes of Health. So those are the 2 types of sponsors that we have. So let me put those on here. You have private industry, which, by the way, as of this video, private industry sponsors are responsible for, I think it was 90% of all trials. And then you have NIH or academic institutions or grant recipients. And, with the new administration, Trump actually, it was pretty famous. One of the first things he did was slash NIH spending. So we're seeing less NIH grant recipients. These are your universities that apply for studies. These are single investigator sites usually, whereas these ones are multiple investigator sites, multiple phases. So that's the sponsors, okay? Now, in order to actually enroll subjects, in order to get a drug, a diagnostic, a medical device, a biologic, anything approved by the FDA, you have to go through the clinical trial process. So this involves phase 1. It actually goes before phase 1. There's preclinical. So they've got to do the preclinical. And in the book, we actually talk about the preclinical. So they got to go through the phases, right? And we'll include preclinical here. Preclinical. These are usually studies done in the lab, in vitro. These are studies that are done after in vitro. They're done on animals, right? And then you get into phase 1, which for the most part, phase 1 are healthy volunteer studies or certain indications like oncology, right? First in human. Then we're going to get into IRBs and how these things get approved. But for right now, I'm going to go through the phases. Phase 1, phase 2, phase 3, and then phase 4. Okay, but before we do that, I wanted to talk about some of the other players in the space, which are the investigative sites. Okay, so you have the research sites. In order for sponsors to go through this clinical research process, and this is like 10 years right here. This could be longer. And this is for one study could be over $1 billion, all right? So they're spending a lot of money on this stuff. In order to get any data from these studies and to move on to the next phase, they have to enroll patients. They have to enroll study participants into the studies. So the sponsors actually can't enroll patients themselves because they don't have access to patients. They need to rely on investigators. All right. So these are investigators, and these also could be synonymous with different things like investigative sites. Research sites, these are all the same word, they're synonymous. PIs. Alright, these are the people that are actually in the clinics that are actually enrolling study participants into these studies. So the sponsors, when they're planning a study and they have a study, let's say after they get past preclinical, when they get to phase 1 or phase 2 or phase 3 and then phase 4, which we're going to get into, it's post-marketing, they need patients for these studies. So they contract with various sites across the country. Sometimes it's just one site, like for phase 1, typically it's 1 or 2 sites. For phase 2, they usually use a couple dozen sites, up to 50. For phase 3, I've seen anywhere from 20 to several hundred sites. And then phase 4 is post-marketing studies, so the drug's already approved. So as they need more patients for these phases, they actually need more sites. So the investigative sites, okay, sponsors contract with them for the whatever phase of study that they're doing. They need to get that many sites, all right? Each site tells them how many patients they think they can enroll in that study. The sites actually do all the work when it comes to enrolling the patients, collecting the data. The sponsors are the ones designing the protocol, right? So this is something very important in research. Every study has one, or else you don't have a study. It's called a protocol. And the sponsors create the protocol, whether they're industry sponsors or NIH sponsor, right? They'll create the protocol. The sites that are contracted by the sponsors to enroll patients and follow the protocol, that's all the sites are doing. They're following the protocol. Nobody is creating their own protocol at these sites. You're not allowed to do that. The sponsor creates the protocol. We're going to get into later what an IRB is and how they have to approve each protocol, right? But for now, sponsors create a protocol. Investigative sites follow that protocol. Follow it to the T, and later on in this video you're going to see what happens when they don't follow the protocol, which happens, it's inevitable to happen occasionally when you're doing a clinical trial. So that's the investigative sites, PIs, sites. Who works at these investigative sites? Well, we already mentioned the PIs, but there's also something called study coordinators or clinical research coordinators. And in the book, we talk about this role quite a bit because the study coordinator is the backbone for any clinical research. Everybody thinks it's the PI, and yes, the PI on paper and the PI according to the FDA, which, by the way, we didn't even put here, but that's the regulatory entity, and we're going to get into the FDA quite a bit, right? So the FDA is the one that, the PI is responsible for everything, the entire conduct of the study. But the one that's actually doing all the work, usually at most of these sites, are the study coordinators. Right, so that's who works at the sites, and then there's other people at the sites too, sub-Is, and we're going to save that for later on in the video because we're going to get into site dynamics. The way the book's actually structured, right now we're just giving a bird's eye overview of research, but we're actually going to get into the specifics at the site level, at the sponsor level, and at the CRO level, which we didn't even introduce now. So let's talk about the contract research organizations, or CROs. So, sponsors, there's something called core competency in business. It means doing what you do well, and only doing that. So, sponsors, they're not necessarily in the business of managing clinical trials. They're in the business of creating molecules, creating new investigational products, which are what they're testing. They're not really in the business. Their core competency is not, let's find PIs, let's find appropriate sites for these studies. Some of them do that now, and we're going to see how the 2016 GCP revisions kind of force the sponsors to do more of that. But for now, just keep in mind, core competency, sponsors are focused on creating the investigational product and then selling it once it's approved. And yes, overseeing it as it goes through the clinical trials, but a special kind of organization evolved over the years from the 1970s, and they're called contract research organizations, or CROs, okay? And CROs are contracted by the sponsors sometimes. They don't have to, right? There's no requirement that sponsors need to use a CRO, but for the most part they do. CROs are being utilized since the 70s. They probably hit their peak in the mid-2000s, late 2000s. There's still a lot of CROs out there. There's still a lot of market share. There's been some concern from people in the CRO space that they've been losing market share due to the 2016 GCP revisions, which we're going to learn about in the book for sure, but in this video later on as well. But CROs are still prominent, and CROs are the ones that manage the study for the sponsor. So unlike the sites where the sponsor only needs, unlike the sites where the sponsor usually needs more than one site, the CRO, the sponsor only needs one CRO for a study. There's no getting 2 CROs or 3 CROs like sites. CROs don't do anything with patients. All they do is paperwork. So we're going to get into what the CROs do. So if a sponsor chooses to utilize a CRO, what the sponsor is going to do is have that CRO be in charge of selecting sites. So the CRO is going to select the sites. The CRO is going to monitor the study. And this is where we get CRAs. CRAs are the biggest, they comprise the biggest percentage of the workforce for a CRO. And then helping collect the data from the sites, right? Because at the end of the day, the product that the sites are giving to the sponsor is data. That's all we're giving, right? The sponsors pay a site to get data. We're collecting data for the study so we can move on to the next phase. So after phase 3, we can get FDA approval, or the FDA may reject it, and we're going to get to that point as well. So that's the dynamics, right? You have sponsors, you have CROs, you have sites. You don't necessarily need a CRO, but most sponsors use CROs, but you don't need CROs. But you need sites. There's no study that can be done without a site, because there's no study that can be done without patients, at least not yet. All right, so the sites have access to the patients. And in order to get data, you need patients to be enrolled in these studies so that you can collect the data and follow the protocols so that you can then move from phase 1 to phase 2, phase 2 to phase 3, and then phase 3 to the FDA for approval. Now, the regulatory agency in charge of overseeing all of this is the Food and Drug Administration, all right? And they have set guidelines, right? They evolved over time, but they're called ICH GCP guidelines, right? ICH stands for International Conference on Harmonization. GCP stands for Good Clinical Practice. So these are guidelines that the FDA put in place for sponsors, CROs, and investigators to follow, all right? At the core of the guidelines is patient safety. So, and later in the book and later in this video, we're going to get into how ICH GCP evolved. But for now, we're doing just the overview. Just know that the FDA is there, and they're the regulatory body that oversees the study. So we talked about the FDA, we talked about the sites, we talked about the sponsors, we talked about the CROs, we briefly mentioned CRAs, even though most of you watching or listening are probably interested in becoming a CRA because that's probably the household name if there is one in clinical research. It's a job that's always in demand. It's a job that is always undersupplied, and because of that, the market and the economics that the market gives is high salaries and stable jobs. So it's natural that you've probably been attracted to this video because you're considering being a CRA or maybe a study coordinator, or maybe you'll learn after watching this video or reading the book that the best way to actually do that is to do it from the site level first as a study coordinator. So, but right now we're just giving an overview of clinical research, a bird's eye view. So there's other players here. These are the main players. You've got the sponsors, you've got the patients, you've got the investigative sites, you maybe have the CROs, you still have them in most cases, but they're not required. You have the FDA, which is the regulatory body in the United States, and then you have the IRB, which we just briefly mentioned. The IRB is the Institutional Review Board. I'm going to write that down. They're also known as ethics committees. Institutional Review Board. Or sometimes they're called ethics committees. Right? Now there's actually some details in regards to IRBs that we're going to get into later. But for right now, just know the IRBs don't care about anything else other than patient safety. They are an independent and neutral party, meaning they don't have any incentive from the sponsor to make sure that their drug or their data looks good. The IRB is an ethics committee that came because of a number of things in history, the Nuremberg Code, the Declaration of Helsinki, the Belmont Report, things that we're going to get into later on in this video, definitely in the book, definitely in the audio book, which accompanies the book where we go off script on a lot of the stuff. The IRBs are in charge of patient safety, patient ethics, right? Yes, you might be asking yourself, why do we need an IRB if the investigative sites, the PIs, as you're going to learn soon, their main responsibility is patient safety as well. First of all, they're physicians, so patient safety should be at their core, but also that is part of good clinical practice, is to do no harm to patients and to keep patient safety as top priority for any study, right? Well, you need the IRB because sometimes the PIs will inadvertently maybe make a deviation or make a mistake that puts patient health in jeopardy and puts patient at unnecessary risk. So the IRB is there to sort of oversee and make sure that the investigative sites are properly staffed and qualified to do a study. But really, let's take another step back. What the IRBs first do is they have to approve this protocol, right? So we mentioned this protocol. You can't do a study without having the protocol. And the sponsors can't just write any protocol they want and then the FDA says, yeah, that's fine, you can run that study. First of all, the FDA is not an ethical body, they're a regulatory body, so they're not in charge of ethics. They might not even know what's ethical and what's not in some situations. IRBs fill that role. So when a sponsor creates a protocol, not only does the science have to be sound, and we're going to get into how that came about, but it has to be ethical. And the IRB has to actually look at the protocol, read the protocol, understand who the patients are that they're trying to target, what the study will involve, does it put the patients at unnecessary harm or risk, do the benefits outweigh the risks for the patients. There's a lot of factors that go into the IRB approving the protocol. So that's probably the biggest thing that the IRBs do, is approve a protocol based on whether it's ethical or not for the patients. So actually the one that looks at the IRB also is the FDA, but it's also the Office of Human Research Protection, OHRP. So they're the ones that actually accredit the IRBs. In the books we actually go through this in great detail, same thing in the audiobook, but just know that the FDA audits IRBs regularly, and so does the Office of Human Research Protection, okay, OHRP. So not only does the IRB have to approve or disapprove a protocol, but the IRB then has to make sure that the patients, and we're going to talk about this soon, are properly informed about the nature of the study. And the patients have to be willing participants, right? And this stems from the Nuremberg Code, which happened after World War II, where the Nazis did studies on prisoners. Obviously, they didn't have to tell them they're in a study. They're prisoners, and the Nazis did whatever they wanted to the patients. And then a lot of the doctors were charged after the war with war crimes, after the Nuremberg conference, where they basically put a lot of these doctors in jail, and obviously came up with the Nuremberg Code, and that was really the foundation of having ethics in clinical research. So patients need to be informed of their participation in a study, be told the benefits and risks, and they need to be willing participants. So the IRB is making sure that all of this is happening, right? Because you can't just rely on the PI to say, or the investigative site, the physician at the site is called a PI. You can't just rely on the PI to say, yeah, you know, we told our patients all the risks, benefits, and they're all willing participants. No, it has to be formalized, and that's done through an informed consent form, which we're going to get into. But then there's other vendors I wanted to talk about. There's other stakeholders in this as well, and one of the stakeholders is, especially now, and they really gained prominence when I started in clinical research around 2005, is EDC, Electronic Data Capture System vendors. So EDC, we're going to get into the other stakeholders now, but these are, what we've covered is the big players, right? So it starts with the sponsors having an idea and then creating a protocol. Then the IRB has to approve that protocol. Then the sponsor needs to start looking at where these patients are going to come from. So usually, not usually, always they're going to come from investigative sites. And then the sponsor needs to decide, okay, do I want to locate all these investigative sites myself? Or am I going to hire the services of a contract research organization to identify the subjects, or identify the sites that will enroll the subjects, monitor the study as it's going, collect the data for the sponsor, and that's primarily how they do it, through EDC systems, which is the main other stakeholder that we're going to go through. EDC, Electronic Data Capture. So these are the main players, and then you have the FDA, who oversees this whole thing, and then you have IRBs, who oversee the safety and welfare and the ethics of the study for the study participants. So that's the landscape, that's the bird's eye view in a nutshell, because oftentimes what I've noticed from my CRA Academy or from people that watch on YouTube and ask me questions about just like very detailed things in clinical research is they don't understand, they don't have a clear understanding of the overview of clinical research, the bird's eye overview. So one more thing I wanted to add in this overview. In order to even do a human study, the sponsor needs to file with the FDA what's called an IND, Investigational New Drug Application. I don't know why they don't put the A in the acronym, but that's what it's called. It's called the IND. So before the sponsor is even allowed to start testing on humans, they have to get an IND from the FDA. So the FDA has to say, okay, you can use this drug, this investigational product, in clinical research. So they can do phase 1, healthy volunteers, phase 2, a small group of the disease population, phase 3, a larger group of the disease population, and then after the phase 3 study, which is called a pivotal study, which we're going to get into later on in this video and certainly in the book, we're going to go into it in great detail. This is the study, this is the phase where after the study, the sponsor submits all the data to the FDA, hopefully to get approval of the drug so that doctors can start prescribing the drug. So what that is called, if they get approval, is called an NDA. Right? New drug application. So that's what the sponsors are trying to go after, is to get this NDA, and it takes years to get to that point. They've been trying to cut these timelines down. Technology's been helping, and we're going to get into how technology helps. But really the bottleneck, because like I started this video out with, clinical research is the bottleneck for innovation, but it's also the source of the innovation. So the bottleneck has always stemmed from low patient accrual rates. So no matter how much we get into technology, there's EDC, electronic data capture system vendors, there's eSource vendors, there's ways to randomize IWRS, IVRS. There's tons of vendors in this space. There's patient recruitment firms, all right, recruiters there's lab vendors. They don't really help with patient recruitment, but a lot of this technology is designed to get this data from these phases analyzed quicker, right? And we're going to learn strategies that CROs and sponsors are using. Risk-based monitoring is one of them, remote monitoring, to where they can shorten these timelines and also reduce the costs that are involved in running a clinical research study. So, research sites, here's where people get confused as well with this overview. And we're not really going to go into many more of the other stakeholders. These are the big ones. You have electronic data capture, you have eSource, you have IWRS, you have labs, safety labs. Technically the IRB is a vendor. And then you have a lot of other things like e-regulatory, which is now, I mean, in a 2019 world, 2019 and beyond, everything's going to be more digital now, right? But that doesn't stop, and that moving from paper to digital does nothing to reduce the bottleneck of getting more patients in studies quicker. That is the problem with clinical research since it got started, and that's going to continue to be the problem with or without technology. And we're going to see how that develops in the book, and like the intricacies and the nuances of that. But right now, one thing people get confused about is with these sites. Sometimes these sites are also universities. And I mentioned here that sometimes universities get grants and do their own studies. So a lot of these times, these universities, these academic institutions, these teaching hospitals, they can get their own grants and actually be the sponsor of their own studies, which is this category right here. These guys are their own sponsors for their study. right? They get grants from the NIH. It could be a university. It could be University of Arizona. It could be Arizona State University. It could be any university. They get grants, usually from the NIH or other similar entities, to run their own studies. But that number is small compared to the industry-sponsored studies, which are about 90% of the trials. In the industry-sponsored studies, there's no grants given by anybody. No government grants given to fund research. Big pharma, biotech, private investors are funding research in the hopes of getting this NDA, right? And getting drug approval, or getting the investigational product, which is the term used for whatever it is that they're studying. It could be a pill, it could be a device, it could be a diagnostic, it could be a biologic, number of things. It's called investigational product. That's what everyone's trying to get, is the FDA approval for that investigational product so that they can go to market and start selling. And the more expensive this stuff is, the higher the price of that investigational product will be once it's approved. Right? So that this contributes directly to healthcare costs. And the bottleneck, again, is the patients. That is the bottleneck. So this is the bird's eye overview of clinical research. I wanted to get into the next section of the book, which is the Comprehensive Guide to Clinical Research. And I guess we can talk a little bit about, there's financial aspects, there's financial dynamics. We kind of covered that a bit. Private industry, the drug companies or biotechs or medical device companies are sponsoring the studies, but none of these players do anything for free. So the sites have to get paid. They usually get paid to enroll patients based on patient enrollment. We're going to get into that in the site dynamics section. CROs get paid based on the work they do. Obviously the CRAs who work at the CROs get paid, and the other staff that are employed by CROs or sponsors, which are usually high-paid people. I mean, these are six-figure plus careers for most of the people that work at a CRO or a sponsor, even at a site level in some cases. So there's a lot of financial aspects. Even patients get paid for their participation most of the time. So there's money. There's a lot of money being spent on this. And last I checked, and the book will have the exact amount, but I'm not reading the book verbatim for this video, it's about $1 billion to get a drug from preclinical all the way to approval. And then phase 4 is post-marketing. So at this point, the drug is already approved, but the drug company or whomever is sponsoring the study wants to get some more data on the drug. So maybe they want to remove an adverse event or a side effect from the medication label, but the drug's currently being marketed. If you're watching any TV channel, you'll see a drug ad, and then at the end you'll see all the list of possible side effects. Right, those are called adverse events, which we're going to get into later in the site dynamics section. But basically each one of those adverse events that's listed, that the sponsor has to and is required by the FDA to disclose, could be something that the drug company decides they want to remove, but in order to do that, they need additional data from the FDA. The FDA will only allow them to remove a side effect from its marketing if they do more studies and they can prove that it should be removed. So that's what phase 4 studies come out. Phase 4 studies are also useful for economic outcomes of patients. So a perfect example is a patient that's depressed, can't work, can't function properly. Now they're taking an antidepressant. Now they can go back to work. That has economic impacts on them. So these are they collect some data in those cases too, so that they can tell doctors, hey, this patient, we did the study, and patients with depression who took our drug are 30% more likely to have an increase in their salary or go back to work, or just whatever claims they could make are required to be made in phase 4 studies. And sometimes if it's a serious thing that the drug company is trying to remove, like a black box warning label, a good example is Zyprexa, which was an antipsychotic. It was known to increase diabetes or the likelihood of whoever's taking it to get diabetes. It was approved The drug was approved by the FDA because it worked for schizophrenia, but one of the side effects was diabetes, weight gain, which eventually led to diabetes. So they had a black box label, and obviously they wanted to remove that because it would sell more if they could remove it. So they had to go all the way back and do more studies to remove that label, and they never were able to do that. So that's the financial aspects of this. Just to give a quick overview, again, in the actual book, we go into great detail on this, and we go off script on the audiobook as well. We talk a lot about this stuff. Okay, so I guess we can talk a little bit about CRAs. I'm going to focus on CRAs and study coordinators. So now that we understand the bird's eye view of all the main players, we are going to talk about CRAs and CRCs. Alright, so before we get into the phases of clinical research, and I'm going to kind of give another overview of the phases. So everything that I'm going through is like an overview. First I gave the overview of the entire research industry, which is what we just did. Now I'm going to give you just a brief overview of a CRC and a CRA, which are the most common positions in this industry. And maybe I'll mention, of course, we're going to mention line managers, in-house CRAs, principal investigators, who all these people work for. But basically they either work for the sites, the sponsors, or the CROs, right? That's it. Like 90% of the workforce in this industry works at one of those three stakeholders that we discussed earlier in the overview. So CRC, let's talk about CRC first, Clinical Research Coordinator. CRC, and then you have CRA. These are by far the most common positions, the most abundant positions in the industry, and I mean, they're both good careers. CRAs get paid more, like maybe double what a coordinator gets, sometimes triple, sometimes quadruple, right? Now, CRCs, this is more of an entry-level position, but even nowadays, it's hard to find an entry-level CRC position. What you'd really find is a clinical research assistant position, which is another CRA, so this is why it's no surprise why people get confused with these things. So, clinical research coordinator, and the roadmap to get to be a clinical research coordinator is an assistant, usually. Sometimes you can get to be a study coordinator. Now remember, study coordinators are employed by research sites, are employed by investigative sites. They're usually small clinics, like the one I'm standing in right now. Sometimes they're big clinics, like university departments, and everything in between, right? Medium-sized, large, university, hospitals, mom and pop sites. They work for the research clinics. They work for the investigative sites, right? So depending on the coordinator's potential, typically you don't even need any bachelor's degree or anything like that to be a coordinator. Although it certainly doesn't hurt to have a bachelor's degree or even beyond, have a master's degree and be a coordinator. I know some PhD people that are coordinators because they want to get to be a CRA. So you don't need that to be a coordinator. You don't need any degree to be a coordinator. You do need at least a bachelor's degree or the international equivalent to be a CRA. Okay? I don't know why. Probably because they get paid so much. Right? An entry-level salary. In the book, we get into the salaries and all that at the end of the book. In this video, I might just go through the salaries briefly. But an average salary for a coordinator, somewhere between, and again, I'm talking averages, somewhere between 45 and 65K a year. Average salary for a CRA, somewhere between 60 to 100 plus. Right? I know some CRAs that make 300K a year, but they're anomalies. Usually it's in this range. Right? depending on the expertise, depending on what kind of studies they have experience with, depending on how long they've been a CRA for, which I guess is expertise, and what part of the country they're in. If you're in California, you're going to get paid more than somebody who's in North Dakota. Same job, right? But these are the ranges, and same thing with the coordinators. So it's no wonder most people watching this video or reading this book are wanting to be CRAs, probably because a friend of theirs is a CRA and they told them how much they get paid, or they just read about it. CNN, Fortune magazine, every year they talk about this position as one of the top 10 most lucrative positions and most stable positions in any industry. So it's no wonder why people want to become CRAs. It's just extremely hard to get to be a CRA without any experience. And there's a roadmap for how to become a CRA, which we're going to talk about briefly in this video, and I'm actually going to include that in the book as well. A roadmap for how to become a CRC or a CRA. So to be a CRC, you can get hired straight out of college with no previous industry experience as a coordinator if the investigative site or whomever's running that site, maybe it's the PI, maybe it's a site director, if they see that this person has potential, they may take a chance. I've done this with many people. I've taken chances on people because I hire clinical research coordinators at my clinics, and I've taken chances on people with zero experience because I can teach them how I want things done at my site and how to follow my SOPs. Sometimes it's better to hand groom somebody who knows nothing about research and to get them to follow your SOPs properly, as opposed to bringing on somebody who's used to doing things a certain way at another clinic, but then that doesn't work in your clinic for whatever reason. SOPs, we're going to get into SOPs later too, standard operating procedures, but sites need to follow standard operating procedures, and sometimes the best way to get somebody to follow your SOPs is for somebody who doesn't know any better. All they know is your SOPs, as opposed to an experienced coordinator from another clinic who's going to bring their SOPs and their way of doing things to your site, which may be good or bad. It could be a good thing too if you don't know what you're doing, or maybe it's an improvement to the way you operate at your research clinic, but sometimes it could be bad too. So all of this to say, yes, it's possible to be an entry-level coordinator with no prior experience. It's less likely to be an entry-level CRA with no prior experience. So it usually requires a longer roadmap to get to be a CRA as opposed to being a coordinator. Now, if you're one of those people that doesn't maybe have potential, or somebody doesn't see potential in you to be a coordinator right off the bat, they might bring you in to be a research assistant. What a research assistant does is help the study coordinator run the study, right? So if you recall from the site, CRCs are hired by the sites and they work for the sites, and CRAs can work for either the sponsor. There's many sponsors who hire CRAs directly and don't use a CRO, or more likely they work for the CRO. Can't work for both at the same time. Okay. Well, I guess you can, but that'd be too confusing for this introductory video. So just know, coordinators work for sites, CRAs work for sponsors or CROs. Right? Now, what does a coordinator actually do at a site? If you remember from the previous overview, sites are responsible for executing the protocol. So the sponsor writes a protocol, selects the sites who are going to follow the protocol, and enroll patients in that study who are then going to be put through the protocol. Right? So coordinators have patient interaction. Patient interaction. Now remember, the PI, the principal investigator of the site, is supposed to maintain oversight, meaning they promise to the FDA, and we're going to see it later in this book. We talk about it a lot in the book, in the audio book, later on in this video as well, the 1572 form and the statement of compliance that the investigator makes with the FDA every time they agree to do a study. Every time they agree to be the principal investigator of a study, the PI agrees by signing the 1572 form that they and only they are responsible for the conduct of the study at their site. Right? Now, that doesn't mean they do all the work, because they don't. In most cases, they do not do almost any of the work other than maintaining patient safety and oversight. Right? And making sure that their staff, who they've delegated, which we're going to get into in the regulatory documents later, much later in this video, and later in the book as well, they make sure that the staff that they delegated are following the protocol properly. And that's primarily the study coordinator, the clinical research coordinator. So clinical research coordinator does most of the work. They're the backbone of the study. This is why experienced coordinators make really good CRAs. And we haven't even really talked about what a CRA does, but we're going to in this overview. So experienced coordinators make really good CRAs, and that's usually a goal for many of these coordinators. Another goal of a coordinator is to become a site director. The site director usually is the site owner or somebody who gets paid more than a coordinator. Maybe they supervise the coordinator. Maybe they even supervise the PIs in some cases. All the PIs have to do is make sure that the patient safety is top priority. They're reviewing lab results. We're going to get into site dynamics later on in this video. But for now, just know this is what a coordinator does. They take the protocol, they get trained by the PI on how to handle the protocol. They ask the PI for any questions they may have about side effects or what's called adverse events, which is a side effect that a patient may have while they're in a study. The PI, they bring the PI the ECG results, the lab results. They have the PI write the progress notes, how the patient is progressing throughout the course of the study. But the coordinator is, like the word says, coordinating all these things. So they're making sure the patients are coming into the clinic as they should. We're going to get into schedule of assessments later on in this video. They make sure that the protocol is that every assessment is being done according to the protocol at every visit, as per protocol, right? The coordinator is making sure that the patients are receiving their medication or the investigational product if it's not a medication. The coordinator is really doing everything, and they're also documenting everything. The coordinator is collecting all this data from their patient visits. As patients are enrolled in the study, they go through visits. The coordinators are collecting all this data on source documents. And we're going to get into source documents later, but it's good to get a brief overview of what a source document is, right? So basically what a source document is any piece of data about a patient, whether it could be medical history. Medical history is a source document. It could be conmeds, which are concomitant medications, meaning what other medication is the patient taking while they're in the study. It's usually not that they're only taking one medication for the study, and that's just the study drug. Oftentimes they're taking other medications which the protocol may or may not allow for. And the coordinator has to know these things, so does the PI. But really, the practical aspects of this whole thing is the coordinator is doing most of this work, right? So these are some of the source documents. ECG results are another source document. ECG. Lab results are another source document. Patient questionnaires are another source document. Vital signs. I mean, who do you think's collecting all this stuff at the site? It's rarely going to be the PI. It's going to be the coordinator. The only thing the PI does is acknowledge that the labs are in order, that the conmeds are there, that the patient is a good fit for the study. These are the kind of things the PI does, but the coordinator actually collects all this data, puts it in the source document, and then it becomes something that's reviewed by the CRA, which we're going to get into right now. SDR, source data review, and SDV, source data verification. So we're going to get into that right now, but I think this is a good overview of what a coordinator does. They basically take a protocol. The PI will accept the study, will train his or her staff on how to do that study, and then the coordinators is one of those staff members that basically takes the protocol and runs with it. So they start enrolling the patients. Yes, the PI is supposed to maintain oversight and make sure that the patients are appropriate for the studies. That's something that's called inclusion-exclusion criteria, which we're going to get into a little bit more, but it's good to have an overview of this. Every protocol has an inclusion-exclusion criteria, also called the IE criteria. And what this inclusion-exclusion criteria says is only patients who meet this inclusion criteria are eligible for the studies, and they cannot meet any of the exclusion criteria. Now sometimes these inclusion-exclusion criteria are really long, and somebody has to really, like when there's a patient that's a possible candidate for being in the study, they really have to go through all the inclusion-exclusion criteria carefully to make sure that the patient is appropriate for the study. The last thing a PI wants to do or a site wants to do is to enroll a patient in a study that does not actually qualify for that study. Because number one, it could be dangerous for the patient, but number two, it's bad data. The sponsor's not going to be able to use that data because that's a protocol deviation right there. We're going to get into what those are, but the FDA will not allow deviations, especially from the inclusion-exclusion criteria. A patient that does not belong in the study, if they accidentally get enrolled, they can't use that data for the FDA. The FDA will not allow that. So the coordinator's with the PI, but really it's the coordinator that's making sure that all these things are going on. And usually a coordinator is in charge of multiple studies. It's not just one study that a coordinator is coordinating on. It's usually multiple studies, and we're going to get into later in the book and in this video some typical work-life concepts, what the appropriate workload is for a coordinator, what the appropriate workload is for a CRA, what's typical. We get into all that, but usually a coordinator is running multiple studies, not just one. Okay, so they're collecting source. The coordinators are collecting source from the patients, right? Anything could be source. Anything that's data that the study needs to capture is source. They're collecting source, and then they're entering all this source into an electronic data capture system. EDC, electronic data capture system. So the coordinators collect the source, and then they enter it in the EDC system, which we mentioned in the last overview is one of the other vendors. Okay. Now, this EDC system contains all the data that is going to be presented to the FDA. Now, the FDA is allowed to audit the sites at any time. Let's put the FDA here in the corner because they should always be, they're always a presence. Even when you don't see them, they're there. They can audit the site at any time for any reason. So anything that's in the EDC system, the electronic data capture system, should be in the source document at one point. And the source document can never be altered. If it's altered, there has to be a reason why. We have to follow ALCOA principles, ALCOA-C, which we're going to get into maybe in this overview. Let's actually get into it now. So ALCOA-C. So let's get into what ALCOA-C actually means. Okay, every industry uses this. It's not just clinical research. It's documentation standards. They're generally accepted in many industries. Clinical research has adopted it, and it makes sense in clinical research. So every piece of data, every piece of source data that we just discussed has to be attributable, legible, we'll define what these things mean, contemporaneous, original, accurate, and now, after the 2016 GCP revisions, complete. They just added, very tough to write that way, they just added complete. And we're going to talk about what each of these things are right now. So, when a coordinator collects source data, all the stuff we just discussed, then they enter the data in the EDC. This source document stays. It stays in a patient chart. Never goes away. The sites actually have to retain these records for many, many years after the study is over. Right? So the source data is always going to be there. The EDC data is also going to be there. That's the electronic version of the source data. It's basically the source data put into a database. So you have both source and EDC. And that is the basis for source data verification, which is the CRA's primary function. We'll get into that in a little bit. Let's go to ALCOA really quick. So the first letter in ALCOA, attributable. Okay? Every piece of data that's collected for any patient visit at any given time in that study has to be attributable to someone. Who collected that data? Has to be clear. Who can it be attributed to? It also has to be legible, and doctors and coordinators too, myself included, look how I write. Sometimes you can't read what I'm writing. Well, anything you collect for research must be legible, right? So someone has to be able to read it and see what it is. You can't just like write in sloppy cursive and say that data is whatever you wanted it to be, right? Contemporaneous means it has to be timely. When was this data captured? When? All right, when was it captured? It has to be, this also means you can't backdate, right? Any data that you collect, you have to put the date that you collected it, and you can't backdate. You couldn't have forgotten to collect something and then a week later go back and say that you got it on that date, right? It has to be contemporaneous. Original, if you have a patient file, which is your source documents, and a patient comes in for a visit, and you give your medical assistant, let's say the coordinator is busy with another patient, so they give their medical assistant, they tell the medical assistant, hey, go collect vital signs for this patient. That's fine, as long as that medical assistant's on the delegation of duties log, which we're going to get into later in this video. That's a very important thing, delegation of duties, delegation of authorities. We're going to get into that. So the MA, the medical assistant, which also is another position that can become a study coordinator, medical assistant. It's the same thing as a research assistant, but MAs, there's schools for MAs. It's not bachelor's, I think it's like an associate's degree, medical assistant. They often do vitals, collect blood. In the case of research, ship the blood samples to the lab. We're going to get into that in site dynamics. So anyways, the coordinator's busy. The coordinator has the source documents for the patient, but they're busy with another patient. So they tell their MA, hey, go be an assistant, go do the vital signs for this patient. The MA goes with the patient, but they don't have the source document, so they write the vital signs on a sticky note like this, okay? Blood pressure, 120/80, weight, height. They actually do all this, and then they record it on a sticky note because they don't have the actual source document with them. That's okay, but that sticky note now has to become part of the source document. And yes, the coordinator can later go in and transcribe the data from the sticky note onto the source document so that the CRA can actually see it, right? But this sticky note needs to be taped on the source document. You can't just throw this away once you transcribe it. Because what if you accidentally transcribe it, or I don't know what the word is, but you transcribed it wrong, right? What if you did that? Then let's say you put 120/80 and the coordinator put 80/120. It's a mistake, right? But then you throw this away. This is the original source. Now nobody knows what the actual blood pressure was. Was it really 120? Was it really 80/120, which would be an adverse event? Or was it 120/80? So this becomes source. This becomes original. This is the original source, right? And you can transcribe it to your actual source, but this needs to stay. This is the original source. Accurate. Back to the 120/80. How do we know if it wasn't 80/120? We don't know unless we have the original source. And then we have to make sure that that's accurate. Is it really what happened? And that's what the CRA's job is to do. source data review, source data verification. Finally, complete. So this is actually a good example, this sticky note with the source document that we just described with the vital signs. Let's say the MA took the vital signs on this sticky note, right? And they kept the sticky note on the source. But on that actual source, the vital signs are blank because it's already on a sticky note, okay? That's technically incomplete because you're having a blank piece of data. Even though you have the original source there, if an auditor were to come in and somehow this got lost, they wouldn't know that it was ever done. So now they added complete. You can't really leave blanks on source documents. You gotta have everything filled in. Wherever there's a spot for something to be filled in, it should be filled in. If there's nothing to be filled in, if it's unknown or if it's not applicable, you gotta line through that data point, line through it, and put NA for not applicable, or unknown for unknown, and then initial and date. Right? So that's what it means by complete. Now, that's a summary of what the CRC does, study coordinator. Now the CRA are the ones, they don't have any patient interaction. Zero. They don't see patients ever. All they look at is these source documents for that study and the EDC system where this data gets entered. So what they do is they do two things primarily. They do more than two things, but for the purposes of this overview, they do two things. They do source data verification, which is a little easier, so we can start there. So source data verification, they will actually look at what's on the source, what's on the EDC, and make sure that they match. So for example, if the vital signs were 120/80 on the source, they should be 120/80 on the EDC for that particular visit. If a patient has a medical history of depression on the source, they should have a medical history of depression on the EDC. And it just goes on for every single data point. That's easy. That's the easy part of being a CRA. That's not why the CRAs get paid big bucks. Right? You can be a high school dropout and be able to do SDV. You can actually be in junior high and do SDV. You don't need education for that. Where the CRAs really make their money is in the source data review. Right? So reviewing this source and comparing it to the protocol. Remember, the protocol is king for studies. This is it. This was approved by the IRB. This was created by the sponsor. This was also approved by the FDA. It needs to be followed exactly how it is. Inclusion-exclusion criteria. Perfect example. But the protocol could be, a site could screw up on the protocol in multiple ways. Those are called protocol deviations. We're going to get into protocol deviations later, but it's pretty much exactly what it sounds like. The site did not do, inadvertently or on purpose, what the protocol said. For whatever reason. Sometimes it's for patient safety, and they're allowed to do that. Remember, patient safety comes first. But it doesn't mean that that's not a protocol deviation. So the CRAs, their main job is to make sure that the sites are following the protocol, and that's called source data review. Here's an easy example. Let's say in one of the inclusion criteria for a protocol, subjects with a BMI of over 30 are not allowed to be in the study, right? Let's say at screening visit, and we're going to get into what the visits are later on in this video. Let's say at screening visit, a patient was 6'0" and 300 pounds. And let's say, and I'm not going to do the math right now, but let's say that BMI was actually 31. So they technically should not be in the study according to the inclusion-exclusion criteria. But let's say the site didn't do the BMI calculations. They just wrote down the height and weight and then enrolled the patient in the study. The CRA is supposed to catch that. The CRA comes out to the site based on the activity that site has every 6 to 8 weeks. So every 6 to 8 weeks, the CRAs go out to the sites, to conduct their SDR, SDV activities, or actually much later in this video and definitely in the book we get into every single type of monitoring visits and what typically is done at those visits. But just know for now, every 6 to 8 weeks they go out to the site based on site activity, and they try to go 2 weeks after the first screening of the first patient or 2 weeks after the first randomization, depending on the monitoring plan, which we're going to get into what that is later as well. All right? So every 6 to 8 weeks the CRA goes out to the site to do source data verification, making sure what's on the source is what's on the EDC, and source data review, making sure that the source that the site is collecting is actually compliant with the protocol, and if it's not, the patient should be withdrawn from the study, and if the site didn't withdraw the patient, now it's a protocol deviation. So there's a lot of cascades, there's a lot of triggers that go on. We're going to get into those too later, but just know that's the CRA's primary function. Really, if you ask my opinion, the CRA's primary function is to make sure that only patients that are qualified have been enrolled in the study. All right? Obviously, that's a concern for patient safety. Making sure that only the patients that are qualified to be in that study are enrolled in that study, and making sure that the site is following the protocol. Because if you get an inexperienced site, or even an experienced site, but they happen to be overworked for whatever reason, they make mistakes. And mistakes sometimes can have pretty bad consequences for patients. Right? I know of a site who gave the wrong investigational product to a patient, and it could have killed the patient. Right? They just missed the number on the bottle, dispensed the wrong medication. Obviously a CRA wouldn't be able to catch that anyways going 6 to 8 weeks out after the visit had occurred, but they can at least check that the site has been dosing properly, they've been collecting source data properly, they've been entering the data properly, right? That's another important thing. All those adverse events, all those side effects that are put on the back of the medication bottles when they get approved, those come from adverse events in the EDC system. All right? So if a CRA missed some adverse events, maybe there were adverse events listed on the source but not in the EDC, okay, then there could possibly be unreported adverse events for that drug. Now there's other layers of protection there in place. It's not just all on the CRA, but the CRA is the first line of defense. And the CRA is also the first line of defense between the site and the FDA. Because remember, the FDA can come in and audit a site at any time, for any reason. Right? So a CRA is going to make sure that any errors that the site is committing, source data review, are we following the protocol? Are we following the inclusion-exclusion criteria? Are we following the schedule of assessments, which we're going to get into later? Are they doing all these things? And if not, they need to make sure that they are. And they need to make sure that these deviations that the site's committing are getting reported to the IRB. Deviations get reported to the IRB. We're going to get into that later too. So that's primarily the CRA's primary functions, right? SDR, SDV. Now to become a CRA, so you can see their job is complicated. I think more complicated than study coordinators. And just like coordinators are coordinating more than one study, CRAs are sometimes monitoring. CRAs are also called monitors. I don't know if I mentioned that. Monitors. CRAs are also monitoring more than one site, and sometimes more than one study with multiple sites. So every 6 to 8 weeks, they're probably at a different site, doing the same thing, making sure the sites are following the protocol, making sure that the EDC, the data in the EDC, electronic data capture system, matches what's in the source, so that when the FDA reviews all this stuff, they see that everything is there the way it should be, and that the site was following the protocol. If they're not, the CRA's job is to make sure that these things get corrected, that the proper steps are taken to make sure that these things get remedied, and they can even train the site and train the investigator on proper conduct of the protocol. we're going to get into training as well later on. So let's go to the roadmap for how to get to be a CRA. So you can see from all this that I just explained why it's highly unusual for someone with no research experience to be allowed to be hired to be a CRA. It just doesn't happen that often. There is one exception, all right? RNs, registered nurses, and it has to be BSN RNs, Bachelor of Science in Nursing, registered nurse. Sometimes, especially oncology RNs familiar with oncology, because oncology studies are always understaffed, unqualified people, qualified CRAs. RNs are usually allowed to be entry-level CRAs, so they can bypass all this roadmap. Why? Because they have a great understanding of medical history, of ConMeds, they understand lab results, they understand ECG results, they understand vital signs. They may be able to know what an adverse event is. That's an unreported adverse event that the site didn't report. A perfect example is blood pressure. Let's say a patient came in, the blood pressure was 150 over 100. The site writes it down the way it is, 150 over 100, but they don't put that it's an adverse event. Well, an RN, and that's a simple example for the purposes of this video, sometimes it's more complex. An RN is going to know, hey, I'm an RN, I know that that's an adverse event, and it should be reported as such. That's one example. Another example is contraindicated medications. A lot of these protocols are designed with excluding certain medications, so patients taking certain medications are not allowed to enroll in the study because if it mixes with the investigational product, it could be bad for the patient. Now, oftentimes that stuff goes, thankfully not often enough, but it happens where patients are enrolled taking exclusionary medications, and that's against the protocol, right? And CRAs, preferably CRAs that understand medicine, can catch that, right? So that's why RNs are usually the only ones, they're the only exceptions to no experience in research, but they get hired to be CRAs. Everyone else, I don't care if you're a doctor in another country, if you're an RN in another country, same thing. You have to be licensed in the United States to be an RN, or to be a CRA with zero experience. So everyone else needs to start either as a coordinator, a research assistant, a medical assistant, or there's assistants to the CRAs as well. They're usually called in-house CRAs. In-house CRAs, or clinical trial assistants, so CTAs. There's a number of different positions. Sponsors and CROs, sometimes they call the same position different things across different companies. Unlike sites, at sites usually it's consistent. At sponsors and CROs, the job titles vary, but basically there's people that are helping, they're a support system for the CRAs. They don't get paid as much, but it's a good, sometimes entry-level position, although even these are hard to sometimes get entry-level positions. I know many people who are in-house CRAs with zero experience in research and then went on to become CRAs. Same thing with clinical trial assistants, clinical trial administrators. Remember, this is all on the sponsor and the CRO side. We already talked about the site side. Now, this thing, the study coordinator. Very common for a study coordinator to be a coordinator for 2, 3, 4, 5, 10 years and say, you know what? I don't actually want to be a site director. I like research, but I want to earn more, but I don't want to be a site director. I don't want to own my own site. I think I'm going to go be a CRA because I know how to do their job and I can double my salary. And they're right. Okay, so this is why coordinators make really good CRAs. And it's a good roadmap if you want to be a CRA and you can't get one of these positions at the CRO because they're harder to get, CRO or sponsor. You need to go at the site level. Go at the site, try to get a position. Try to get an entry-level coordinator position, try to get a research assistant position, try to be something we haven't discussed, but I've talked about how patient recruitment is the bottleneck, right? The reason why studies go on, there's study delays. In the book, we give stats on what percentage of studies are delayed. Most studies are delayed and under-enroll and underperform because sites are not accruing patients fast enough. And so this is why medications cost more, because it takes longer to do the trials. It costs $1 million a day for a sponsor, $1 million a day for a sponsor to run a clinical trial. It's expensive stuff. So literally, if a study is delayed by months, I mean, we're talking like 30, 60, 90, $120 million just for that one study. They sometimes have multiple studies before a drug gets approved. So, patient recruiter is another job that sites can hire for. Every site needs help with the recruitment. It doesn't matter what they say, it doesn't matter how big their database is. You can never have enough studies or enough patients for the studies that you have. Because the recruitment, there's a reason why patient recruitment and most studies are delayed, and that's because of lack of patient recruitment. So if you can't get any of these positions and you want to become a CRA or you want to become something else in this industry, one of the best places to start is patient recruitment. And you work for the site in these cases. I did a YouTube interview with someone who got started just this way. Patient recruiter, then she became an in-house CRA. I think she's doing like some kind of high-level stuff at a CRO, feasibility analyst. Right, you could watch that video on YouTube if you want. So patient recruiter is a great way to get started at a research site. It may pay commission only, but that's the price you gotta pay if you want to get into this industry, which is very lucrative, very stable, tons of upward mobility. I mean, all it is upward mobility, right? Even CRAs, once they've been CRAs for a while, they become a lead CRAs. It's another thing we need to talk about. The lead CRAs. Sometimes they're also called the line managers. They are individuals at the CRO or sponsor level that supervise the CRAs, and they make sure, and multiple CRAs, okay, they supervise them, and they make sure that what the monitors are doing, first of all, they make sure the monitors or the CRAs are actually doing their jobs. How do we know that they're actually doing SDR and SDV? It's too late to wait for the FDA to find mistakes. Then it's too late. How do we make sure that they're not slacking? CRAs are busy. It's a stressful job. How do we know that they're catching everything? Line managers. We're going to get into CRA dynamics, monitoring visits, monitoring reports. But when CRAs go to sites to conduct SDR, SDV, and also look at the investigational product dispensing, we're going to get into this during all the breakdown of all the different monitoring visits. They have to write reports. The CRAs have to submit reports and send it to their line manager, and their line manager needs to look through everything in their report and make sure that the CRA is doing everything that they could be doing to keep the FDA happy should the FDA ever audit that study. So that in a nutshell is the overview for the two most common positions, the roadmaps to get there, and then all the support positions and what those individuals do with ALCOA-C explanation, as well as a protocol explanation, a little bit of salary, and we're going to get into more detail later on, but the next module we're going to talk about something else. So today we're going to talk about the different phases of clinical research. There's 4 phases of human clinical trials, and it's phase 1 through 4. So we're going to start talking about it right now. So in phase 1 is the healthy volunteers, and usually there are between 20 to 100 subjects enrolled in these studies, and it's typically done at only 1 or 2, maybe 3 sites. So total patients is between 20 to 100 patients. Now, these are going to be healthy volunteers. Okay, so usually these are healthy volunteers. There's also exceptions for oncology. And in the book, we will speak more in the book about why oncology is an exception for phase 1. But it basically stems around the fact that for oncology trials, they're using chemotherapy. And to give chemotherapy to a healthy volunteer, I believe would be unethical, and most IRBs won't approve it. So for oncology, since the argument is these patients actually need this treatment, they're going to be given, they can do phase 1 studies directly on the cancer patients themselves. Another thing with phase 1 studies is they're often looking for the maximum tolerated dose. So that is called MTD, maximum tolerated dose. Now, they don't do that in oncology. In oncology, they give the chemotherapy. They do sometimes look at what dose ranges work better and when do they start producing side effects for the subjects. But they're really just looking for establishing that maximum tolerated dose. So let's, in the book and in the audio book, we really focus on the exception here. We kind of go off topic a lot. For the purposes of this video, I want to keep it short. Just know that phase 1 is primarily done on healthy volunteers, and what they're doing is they are looking for the maximum tolerated dose. Okay, so they're literally, let's say it's a drug for asthma. They are taking healthy volunteers between 20 to 100 at about 1 to 3 phase 1 clinics. And this is why it's very tough if you're a research site and you want to compete in the phase 1 clinic space. It's really tough, as you'll see in the phase 2, 3, and certainly in phase 4. It's much easier for sites, there's more opportunities for many sites to participate in clinical research. In phase 1, your competition is every phase 1 clinic in the country that can do the study. So it really boils down to having a relationship with that sponsor. But anyways, so the maximum tolerated dose, typically these studies last for a few days up to a couple weeks, okay? So a few days. I've seen phase 1 studies as long as 8 weeks, right? And during this phase, all they're interested in is what's the maximum tolerated dose, at what dose do we start seeing side effects, and what side effects are those, and then what side effects occur at lower doses. They just keep going up until patients literally, and they're healthy volunteers. All right, that's very important to understand. There is no benefit for being in a clinical trial for a phase 1 study for a healthy volunteer. I mean, there's no therapeutic aspects of doing this study. It's all financial gain for them. They get paid a couple $1,000s for their time while they're in the study. And they do have to be healthy. Generally, they have to be within a certain age range, typically 18 to 45, and they have to be in perfectly good health, like no issues with labs, nothing. So they test the IP, they keep raising the dose until side effects start occurring, and then they take them off of the dose. So these are inpatient studies. The patients don't go home. They stay in a unit because there could be potentially they're literally raising the dose until they start experiencing side effects. So they are keeping them in the clinic, in the unit, in the phase 1 unit. All right. Okay, so phase 2 studies, this is the first time that the actual drug, the IP, let's use the example of asthma IP, investigational product. This is the first time that the drug is actually used, again, with the exception of oncology, but don't worry about exceptions right now. This is the first time that the actual IP is being used on the targeted population, so on patients who actually have asthma. Okay, typically they enroll between 100 to 300 patients, all right? And these studies are done on the indications that the IP is supposed to or hypothesized to work on, all right? They are efficacy, which means does it work? Okay. It's the first time any efficacy is being measured in a drug. Remember, here in the phase 1 for healthy volunteers, it's all about safety. What are the drugs? What is the maximum tolerated dose? At what doses do side effects or AEs begin to occur? Here, yes, in phase 2, safety is a priority. It's going to be a priority in phase 3 as well. We're going to get to that. But efficacy is the first time efficacy is being looked at, because it's supposed to work within this dose. So in the phase 1, they find the dose range for healthy people. They say, okay, this particular IP, healthy volunteers start experiencing adverse events at this dose. So we're just going to go underneath that dose, right? And we're going to assume that it's also going to not produce those AEs in these patients, in the patients who actually have the medical condition. So they're using that dose that was established in the phase 1 study. So you can see how being in a phase 2 trial, if you're a study participant, is actually, infinitely safer than being in a phase 1 trial. In a phase 1 trial, if you're getting the study drug, you are going to get side effects. If you get placebo, obviously not. Here, in phase 2, they are also getting placebo or the IP, and those are double-blind, and we're going to get into what double-blind and randomized means later, but actually we can get into it now. So double-blind, any study, for the most part, the FDA considers the gold standard of clinical research to be double-blind. That means the IP could be either the investigational product, placebo, which is an inactive pill. It's like taking a sugar pill. It doesn't do anything. All right? Although there is something called a placebo effect, which is interesting because it means that our minds affect our health in some cases, but we won't get into that right now. And also sometimes there's a third arm in these treatments. It's a different dose of the IP. So that IP, subjects can be randomized on various doses. They can get a placebo, and sometimes they can even get another treatment that's available on the market. That's called a comparator treatment. So studies are designed in different ways. The FDA meets with the sponsor. They determine how the study should be designed for the most part. They give them a roadmap. They give them a framework for how what they would want to see. So they get randomized. Randomized means they're randomly assigned to one of those arms that I mentioned. And we're going to get, in the book we go in great detail, in the audiobook we do. Later in this video we're going to talk about double-blind and randomized and exactly all those things. But for the phases, just know patients will actually be randomly assigned a specific treatment and a specific dose or a placebo, and the patient won't know what they're getting, and neither will any of the study staff for the most part. Okay, those are double-blind, placebo-controlled studies, which is the FDA gold standard, right? So 100 to 300 patients, the indications, okay, they're looking at safety primarily, and then efficacy. So again, they already know the maximum tolerated dose from phase 1. They already know at what dose levels adverse events start coming into play for healthy volunteers. So now they're looking at slightly lower doses for the targeted population. What AEs are occurring there? Okay, and these are usually between 20. I've actually seen 10 to 30 clinics for these studies. 10 patients per clinic would give you 100. At 30, 10 patients per clinic would be 300 patients. Those are about typical numbers for phase 2 studies. And then they also look at efficacy. Okay. Does it actually work at that dose for this amount of time, which typically they last a few months, so anywhere from 4 weeks to, which is 1 month, to 6 months. All right, that's typically the duration of a phase 2 study, and they're looking between 10 to 30 clinics. So that's phase 2. And during this time period, however long the study is, 4 weeks or 6 months or anywhere in between, they're looking not just for safety. All right, because remember, in the phase 1, They're just establishing maximum tolerated dose. And it only goes up at the most to 8 weeks. So here you go longer. You go longer to see, okay, what happens, what starts happening in month 3, month 4, month 5, month 6. And then as far as efficacy as well, does it actually work? We have enough time to see if there's actually signs that it works. All right? If they like the data, if it's promising enough, they move on to phase 3. Now what phase 3 is, these are called pivotal trials. So these are the last studies. Sometimes there's various phase 3 studies for the same IP. Sometimes they change the protocols a little bit from one study to the next, but they're all in phase 3. Right? Sometimes sponsors can afford to run multiple trials at the same time and then see what works best and then send that to the FDA. So phase 3, they require a lot more patients than phase 2. Sometimes it's between a couple hundred to a couple thousand patients, right? It's also on the same indication, but here it's open, and it's something I forgot to mention in the phase 2, so let's do it. So like I mentioned, in our example, the indication was asthma, but it really could be any indication depending on what the IP or the study is testing. In phase 2 studies, the inclusion-exclusion criteria, which we're going to learn about later on in this video, later on in the book, later on in the audiobook, it's the inclusion-exclusion criteria is a part of the protocol which, exactly what it sounds like, dictates what kind of patients are allowed to be in the study and what excludes the patients from being in the study. So a lot of times in these phase 2 studies, because not much is known about how this drug will affect patients with the medical condition, they're very restrictive on the inclusion-exclusion criteria. So you're not really getting an accurate sampling of real-world patients with asthma. You're getting cherry-picked patients with asthma that meet the tight inclusion-exclusion criteria. Okay, they do that for a reason. They want to see safety. Again, they're concerned with safety. So they want to make sure there's not any other medical conditions that could affect the patient and create more AEs and SAEs, which are adverse events and serious adverse events. Okay, so they want to make sure that the patients are patients with asthma, but they don't have a lot of the other medical conditions that other people have in the real world. All right, so it's not really indicative of a real-world patient with asthma, but at least it's somebody with asthma. Okay, in phase 3, they open up the inclusion-exclusion criteria to reflect real-world patients with asthma a little bit more than phase 2. And the FDA understands that there's a risk of cherry-picking subjects, and the FDA in phase 3 will really look at that. What's the IE criteria? How close is this patient, this ideal patient that you're looking for a phase 3 study? How close do they reflect a real-world patient with asthma in the real world who didn't do your study? Because you can't just cherry-pick patients. So in phase 3, they open it up to more patients, between 300 to 1,000. They are always still looking at safety. AEs, SAEs, but they are really looking at efficacy over a longer period of time. So typically a phase 3 study goes on for 6 months to, I've actually seen like 5 years in some studies. That's not typical. Usually it's 6 months to 18 months. But I've seen these phase 3 studies, and again, the FDA, it depends on the indication, it depends on what studies have come out before. Anybody can go on clinicaltrials.gov and look up any indication and see how the studies were designed in the past. So the FDA uses those as roadmaps for how to design your study in the future. Okay, so 6 months to 18 months, or up to 5 years, they're really looking at the efficacy, and this is a long enough period of time to be able to get a good gauge on efficacy. And again, it's double-blind, it's randomized, the gold standard typically, right? Now, after one of the unique things about phase 3 studies is there's something called, usually, something called an open label period. So what this means is, just like I mentioned, studies are double-blind, randomized, placebo-controlled. Patients could be receiving a placebo, and they don't know. Once the study is over for that patient, let's say it's 18 months have gone by, and the patient finished the study, completed the study. And we're going to get into different ways subjects can complete or withdraw from a study later on in this video, and certainly in the book as well. Let's say 18 months has passed, and the subject is done with the study. They completed the study. There is now an open label phase where it's like almost like a new study for that patient. It's called a continuation or an extension of the protocol. You have a completely different set of regulatory documents for that study, a new informed consent for that study, but it's the same study. The only difference is now you're guaranteed the IP. Right? You're guaranteed the investigational product. The open label period is no longer placebo controlled. So if a patient has been on placebo the entire 18 months, they first of all could withdraw at any time during that 18 months, but let's say they stuck with it, their AEs, and somehow their disease wasn't bad enough to where placebo didn't matter, they managed just fine. Well, now in the open label period, and they could probably guess that they were on placebo, and so could the researchers. And matter of fact, that's one of the things that the sponsors look at is, does the IP separate from placebo? Meaning, do the people who received the investigational product, do they respond differently than the people who received placebo? That's called separating from placebo. If it does not separate from placebo, the data is not considered good. There's no signs of efficacy, right? It's no different than receiving nothing. That's not good. But let's say a patient who received the IP or received placebo completed the 18 months period. They didn't know what they were getting, but they can kind of guess. Well, now in the open label period, if they choose to join the open label period, they are guaranteed the investigational product, and they may even be guaranteed a certain dose that they're getting it. Or that part might be blinded. Right? But they're guaranteed the open label, the actual treatment, and that can go on for up to 5 years. So here, where I mentioned 6 months to 5 years, typically the double-blind, placebo-controlled goes like 1.5 years, and then the open label can push it out to 5 years. All right? And then they can really start looking at long-term efficacy and long-term safety and safety of that study drug. All right. So these are the phase 3 studies are called pivotal trials because this is the last study prior to the FDA deciding, are we going to approve this drug? Are we going to grant an NDA, which is a new drug application? So the sponsors package all this data from the EDC. Biostatisticians look at all the data, they package it for the FDA, they present it to the FDA, and the FDA will determine if they're going to approve that drug, and if it's now available for any physician to prescribe it on the market. And this is where the drug companies really start to recoup their investments from all these things. Remember, these are like years. Okay, the study may only be 8 weeks in phase 1, may only be 6 months in phase 2, it may only be 18 months in phase 3, but the enrollment windows to accrue all of these patients takes the site more than 8 weeks, takes the site more than 6 months. Right? To accrue 300 patients, the enrollment window, meaning that the amount of time sites are allowed to enroll patients until this number is accrued across all sites, is much longer than the actual duration of the study for the patient. Those are two things people get confused on, is the study duration for a patient versus the study length for accruing, for all the sites to accrue cumulatively the number of patients needed for that particular indication. And obviously the more patients required, the longer that takes. So this is $1 million a day for a study, for a typical clinical trial. So by the time the drug gets approved, and by the way, many drugs don't make it even this far. Many drugs fail at phase 1, many more fail at phase 2, and many fail at phase 3. So by the time you get those approvals, you can understand why medication costs so much. And we haven't even really gotten into the source data review, source data verification. We've got into it a little bit in this video, but we're going to get into it a little bit more in the site dynamics and the monitoring section of this video and of the book as well. And then phase 4 studies, the drug is already on the market. So these are post-marketing studies. The drug is already on the market. These are usually not placebo-controlled or randomized. These are open label. It's essentially an open label study. And these are also called observational studies. And they could last, sometimes they accrue over 5,000 patients. Typically it's over 2,000. But I've seen as much as 100,000 for some studies, which is unbelievable. Observational, and it's open label. And they're just looking for more marketing data. Like, what is the actual safety profile? What are the financial benefits to the patients, the economic benefits to the patients? They give the patients a lot of surveys, such as satisfaction of life, quality of life, workplace improvements. They're trying to find, the sponsors are trying to find any marketing, any benefits that they can give to their sales reps. Because remember, at this point in the phase 4, the drug is already approved. The drug's already on the market. So they just want to give their sales reps more data, more ammunition, so when they go to doctors' offices and say, hey, our drug does this, this, and that, you know, based on our most recent phase 4 study, it makes it easier to sell. So that's phase 4. This is why it's very easy for brand new research sites to participate in these studies, the phase 4 studies, because they're usually less restrictive. I mean, it's almost like anyone with this indication can join the study. They're guaranteed the study drug. The protocols are very simple compared to phase 3 and phase 2 and certainly phase 1. Protocols get more complex in each phase. So by the time they get to phase 4, it becomes the opposite. It's very easy. Anybody can do it. This is why they're okay with the research-naive physicians to do the clinical trial for phase 4. So if you're a brand new research site or brand new investigator, this is probably the best way to get experience, is to do a phase 4 study. Okay, so now we're going to get into regulations, and there's so much to talk about here, probably just as much as there is to talk about source, which is going to be the next clip after this. But regulations in clinical research, obviously, it's a very highly regulated industry, as it should be. I mean, you're dealing with human lives, you're dealing with safety. So the FDA is the one that, they're the regulator, the Food and Drug Administration. They are the regulator that oversees clinical research, right? They also have other responsibilities. They provide veterinary drugs. They have food safety oversight. So it's not just drugs. It's actually most of the FDA is dedicated towards food, protecting the public from protecting the public and keeping the public safe when it comes to food supply, what we eat. But drugs is a big part of it. And for clinical research, that's really what we're going to focus on. So just know that the FDA, Food and Drug Administration, is the regulator, the primary regulator for clinical research. Okay, so now what's underneath the FDA? There's something called good clinical practice. You may have heard about this. GCP, good clinical practice. And GCP really stemmed from ICH, which is International Conference on Harmonization. So let's go through, and in the book we list all these things and more in the book. There's actually 13 principles of ICH-GCP, which is International Conference on Harmonization. And I'll read them out to you. There's the ethical conduct of clinical trials. So ethics are the foundation of all clinical trials. We're going to get into the Nuremberg Code, the Declaration of Helsinki, and the Belmont Report to kind of give you some background as to how GCP evolved over time. Benefits justify risks for trials. So trials should not be undertaken if risks outweigh benefits. Right safety and well-beings of subjects prevail. Non-clinical and clinical information supports the trial. The protocol must be scientifically sound, detailed, and here's the part where CRAs and sites, this is where they earn their money. Protocols must be scientifically sound and detailed. Okay, that's sponsor responsibility. But here's where it gets juicy for the CRAs and for the sites. And strictly adhered to. Okay? That's the main thing. That's the main scope of an audit. That's the main scope of a CRA. And that should be the main focus for any site, is adhering to the protocol. Alright, IRB approval. We're going to get into institutional review boards, how they came about. Medical care and decisions to be made by a qualified physician. Now, understand, a PI does not have to be a physician. Typically they are. In most cases, PIs are physicians. But there are some exceptions, but that doesn't mean that qualified physicians are not going to be involved in the study. They should be involved at each site. Each individual is appropriately qualified to perform their tasks. We're going to get into delegation of duties log. Informed consent. It's a huge aspect of regulations. All clinical trial information requires accurate reporting, interpretation, and verification. The confidentiality of potentially identifying records of subjects should be protected. This is a big thing when it comes to taking measures to de-identify patients. Investigational products should be manufactured, stored, transported, and distributed according to good manufacturing practices. And in the book we have references to all these. And finally, systems to ensure quality during every aspect of the trial. So this is where CRAs come into place. Right? This is why CRAs get paid the big bucks that you want to get paid, probably, if you're watching this. Right? So there are several countries that put ICH standards into law. The United States uses the ICH standards as part of their good clinical practice, good clinical practice guidelines. Okay, so according to the FDA, GCP is defined as a standard for the design, conduct, performance, monitoring, auditing, recording, analysis, and reporting of clinical trials. So let's get into how all these things actually got put into place, how these things evolved, right? Because the book and this video is going to attempt to blend the academic with the practical, and this is now academic, so we're going to get into exactly how this came about. All right, so there's three and really four pivotal moments in history that kind of helped define and kind of helped mold good clinical practice in what it is today. Okay, the first one is the Nuremberg Code, and this happened in 1947. Now this stemmed from the atrocities that the Nazis committed on prisoners in the concentration camps. As many of you know, or maybe you don't, the Nazis actually conducted human experiments. Obviously they didn't use informed consent. Obviously it wasn't voluntary by the study participants, which were the prisoners. So now there's actually rules that state human research cannot be done on prisoners. That stemmed from the Nuremberg Code. But also what really stemmed from the Nuremberg Code, this happened after World War II ended, where they had the Nuremberg trials and physicians were prosecuted for committing unethical research on unwilling participants. So the Nuremberg Code of 1947 was put into effect. Some of the important principles that came out of this were informed consent for study participants. Right? So informed consent, even though it wasn't formal, there had to have been some kind of informed consent for study participants to understand what they were getting into, and there had to be an absence of coercion, and there had to be beneficence for study participants. There had to actually be some benefits for them. Okay, there had to be some benefits, and the researchers, really the PIs, had to make sure that the risks were not outweighed, or the risks were outweighed by the benefits. So there's more benefits than there are risks to joining the study. Of course, no study is without risks, but they made sure that the benefits outweighed the risks when it came to actually joining a study. So there's actually in the book, there's a full list of principles from the Nuremberg Code as a reference, but just know those are the main important things that came about with the Nuremberg Code. Then in 1964, the Declaration of Helsinki came out. And what the Declaration of Helsinki is widely regarded as the cornerstone document for ethics concerning human experiments. So it built further upon the Nuremberg Code, further pushed for the well-being of the study participants as taking precedence over all other aspects of the study, and the protection of study participants' rights and the protection of vulnerable populations, such as children, as well as the mentally disabled, were key initiatives pushed to the forefront of the Declaration of Helsinki. And this was created by the World Medical Association and is still used to this day to morally bind physicians towards providing a higher standard of protection for human research subjects that the Nuremberg Code called for. So vulnerable populations were protected by this. And what we really had was assent. Okay, so there's informed consent for most people who participate in the trial. If they're mentally capable or if they're adults, they can make their own decision as far as joining a study. They can give consent. But what do you do? How do you protect the people that can't? You know, like parents of the kids need to give consent, but the kids need to understand what they're getting into as well in simpler terms that they can understand. This is called assent. Okay, so that's one of the things that came about because of this. Same with the mentally disabled. They may not be able to understand the informed consent, so they may have a legally authorized representative or a loved one give consent, but they have to demonstrate to the PI that they actually understand the basics of what they're getting into, and they have to give assent. Okay, and then the third thing, the third pivotal moment in history was the Belmont Report of 1979. And the Belmont Report came about, it's really interesting and unfortunate how it came about, but there was something called the Tuskegee Incident. Okay, the Tuskegee Incident was a study, 40-year study, which started in 1932 and it ended probably right before the Belmont Report. So it was a 40-year study. It was completely unethical. They enrolled 600 black males, okay, 399 with syphilis and 201 without. So it was black males only enrolled for this study. 399 with syphilis, 201 without, were enrolled in a study without informed consent, nor were they made aware that they're actually in a study. Okay, these study participants, unknowing study participants, were told by physicians that they were being treated for bad blood. But in actuality, what they received were medical exams, free meals, and burial insurance, but they were never told they were in a study. The most shocking thing about this whole thing, and remember it's a 30-year study, actually 40-year study, when penicillin became available in 1947 for the treatment of syphilis, and remember all these black males who were enrolled, 399 of them had syphilis, 201 did not, and they were just observing, and who knows what else they were doing, but they were primarily observing the progression of the disease. When penicillin became available in 1947, it was not administered. Okay, completely unethical. They could have been given penicillin. they were not, and this posed tremendous unnecessary risk, not just for the men, but also the wives and the children. All right, these are human beings who had families, and this actually paved the way, this incident, the Tuskegee incident, paved the way for IRBs to approve and inform consents to explain in layman's terms what all the risks, benefits, and study alternatives are available to the subjects. So the Belmont report called for benefits for the patients, protection of vulnerable populations, and here's the, probably in my opinion, the most important thing, the most practical thing that the Belmont report actually called for was IRBs need to approve informed consents, and they need to be formalized. And clinical research cannot be marketed as treatment under most, if not all, conditions. Like, patients have to know what they're getting into. All these patients were misled into thinking they were getting treatment, when in reality they were not getting treatment, they were just being observed. Okay, and a lot of lives could have been saved, and a lot of people could have been, there's even generations now, to this day, I think there's like 4 families that are still suffering from the effects of this study. So just, this was a pivotal moment for IRBs, for informed consent, for ethics, for the classification of research as being research and not treatment, and you can't market it as treatment, right? And then in 1981, you had the Common Rule. 1981, the Common Rule. Hopefully you can read that. The Common Rule came into law as Title 45 CFR Part 46, and in the book we actually list out all these Code of Federal Regulations. This mandate provided requirements for how IRBs and their rosters must be structured, and also defined key terms such as research, human subject, and minimal risk. So this actually, the Common Rule, I mean, all these things are the foundation for this good clinical practice and International Conference of Harmonization, right? Which is sort of like it's sort of like NATO, I think, if you know what NATO is, for research, right? And so the Common Rule started regulating IRBs. How the IRBs, you can't just say you have an IRB, you have to get accredited, and that accreditation is through the Office of Human Research Protections, OHRP, Office of Human Research Protections, which oversees IRBs and accredits them, and so this is what the Common Rule brought into effect. So all these things, these are really the pivotal moments for, and the framework for how good clinical practice came about. And there's a lot more about GCP that we're going to talk about later in this video and in the book, of course. And it's primarily around the 2016 revisions to good clinical practice. So just like SOPs, standard operating procedures for sites, which we'll learn about later on in this video and in the book, GCP sort of gets updated as the times call for it. So they just had a very monumental update in 2016, which was, I guess I could talk about it briefly here. We talk about it more later on in the video. But in 2016, GCP revisions basically mandated that it actually focused a lot on electronic systems. So electronic source had to have an audit trail for the electronic source, and I think most importantly, it put a lot of the responsibilities, the FDA put a lot of the responsibilities of the study on sponsors. And sponsors can no longer just outsource to CROs. They could outsource to CROs, but they can't outsource their responsibilities. The FDA, the revisions made it clear that the FDA is going to hold sponsors responsible for the conduct of the study. This means how they're monitoring the accuracy and validity of the study, how they're creating their protocols, how they're managing their vendors, how they're managing their sites, how they're training their people, all those things, how they're looking at monitoring reports, how they're looking at whether the CRAs are doing their job or not. All these things are now their responsibility. They always have been, but now it's written in GCP. Sponsors are responsible for the conduct of the study. And this is why we're starting to see a lot of sponsors bringing a lot of these activities back in-house, meaning yes, they're still using CROs the majority of the time, but their sponsors are starting to get more actively involved. So now that we've learned some of the history of good clinical practice and sort of what the foundation, how the framework and foundation for research as we know it was laid out, let's get into some of the practical aspects, which would probably be helpful for you. But the history, just I wanted to give you some kind of context. In the book, we give you more. So obviously, we just talked about GCP, good clinical practice. And I guess this part of the video is going to be considered regulatory. And we're going to get into what regulatory actually means as a practical matter for research clinics and for CRAs and for coordinators and for PIs and really for anybody that's involved in clinical research. So the first thing is the GCP that we've discussed. There's actually training, and this training should be done by every staff member at a research site, at a CRO, at a sponsor who's involved in any aspect of clinical research. They should do a yearly training of GCP, and they should have a certificate for this. Like, it needs to be on file should an auditor ever come in and look at the site files or CRO files or sponsor files. This means everybody, PIs, CRAs, coordinators, consultants, medical monitors, the list goes on and on. We're going to get into the specifics of what these people do. We've already talked about CRAs and CRCs and PIs. We'll talk about some of the other positions later in the video, and in the book there's a chapter at the end of a whole bunch of positions that maybe you may not have heard of in the past, right? So GCP is really first. Then, for the research sites at least, there are two things that we can just get out of the way quickly. There's a CLIA, which is a Clinical Laboratory Improvement Act. It's not important to know what it is. In the book we talk about it, but just know that every site needs to either have a CLIA or a CLIA waiver. This gives the site permission to draw blood, draw biological samples, process lab samples, and then ship them out to the sponsors or the central labs or their local labs for analysis. So that's pretty simple for a site to get. It doesn't cost that much, but every site needs one, either a CLIA certificate or a CLIA waiver. The next thing is IATA. Okay, IATA is, it's not even really important what it stands for, but it stands for International Air Transport Association. So remember how I said here about shipping labs? It gives sites the opportunity to collect labs, process, ship. Anyone at the site who's involved in shipping or packaging labs and then shipping them out, lab samples, needs to have IATA certificate. So those are easy. I told you they'd be easy. We get them out of the way. Now there's two aspects of clinical research: regulatory. The first one is regulatory startup. So regulatory startup is exactly what it sounds like. Prior to a study actually getting started, all right, before the site is activated, and we're going to talk about what that means too. But when a site is being considered for a study, the sponsor sends out a monitor to do a site selection visit. Prior to the monitor going out for a site selection visit, the site receives a feasibility questionnaire. I'm going to save that for the next component of this video. Later on, we're going to actually go through every single monitoring visit and break them down. But for right now, know that there's a regulatory study startup, and then there's a regulatory maintenance. So, before the site receives IRB approval and before they're ready to start screening subjects at the site, they have to complete the initial regulatory packet. These are also called essential documents. So this is what needs to be collected these are regulatory startups. Before the site is given the green light to screen subjects, and we're going to get into, again, I'm going to break down every single monitoring visit later on in this video. But before the site can actually get the green light to start screening subjects, they need to submit to the sponsor, and in the case of a 1572 form, to the FDA, certain documents which will be considered essential documents. That's what they're called. Okay, so the first one is a CDA or an NDA. Alright, Confidentiality Disclosure Agreement, Non-Disclosure Agreement. Before the site can receive any information about the study, they have to sign a form that says they will not share this confidential information with anyone else other than study staff, appropriate study staff. So that's easy. Then you have the 1572 form. In my opinion, this is the most important regulatory document in research, because all the stuff we just learned about in terms of good clinical practice and ethics and maintaining oversight of people doing the study at the clinic, that is all listed on the 1572 form. And the PI, only the PI, the principal investigator, they're the only ones who actually sign this form, date it, and then they send the 1572 form to the FDA. It's essentially the contract that the PI has with the FDA, stating that he or she understands no matter who they delegate to do what at their clinic, the overall conduct of the study is no one else's responsibility except the principal investigator's responsibility. So this 1572 form stays with the site in what's called a regulatory binder, or reg binder for short. A copy also goes to the FDA, and a copy, sometimes another original, goes to the sponsor on file. And the sponsor, we'll get into how the sponsor keeps these files, but let's first talk about how the site does, because it makes a lot more sense once we get into that. So there's the 1572 form, there's the CDA, there is the actual protocol, of course. So the site should receive the protocol. Now, how does the sponsor know that the PI read the protocol? There's something called the protocol signature page. And it's usually a one-page that says, I, the investigator, received the protocol, I read it, I understand it, and I'm going to train anyone who works for me on the protocol, on the most recently updated protocol. So they sign the protocol signature page. There's also another thing called the investigator brochure. The investigator brochure is all known information and data about the particular investigational product. So remember when we went through all those phases of clinical research, all the data they collect up until that point is included in the investigator brochure. It's really for the PIs to look at. It's a lot of hard science. CRAs have to acknowledge that it's there in the reg binder. Coordinators typically file it in the reg binder, but it's really for the investigators and any other sub-investigators who may be interested in the hard science aspects of the investigator brochure. Investigator brochure is also those little things, those little inserts, the package inserts that you get whenever you buy medication from the pharmacy. They'll give you those inserts. That's like a condensed investigator brochure, but in a study, the investigator brochure is similar to the protocol as far as size and layout. So just like the protocol for the investigator brochure, there's an IB signature page. Right? Another component of the startup regulatory is financial disclosure forms. So on the 1572 form, besides it being a contract that for the FDA and the PI to agree that the conduct of the study is the sole responsibility of the PI, there's also other components, and in the book we actually list the sample 1572 form. But you can just Google right now 1572 form and pull up a PDF of a 1572. Those are filled out. They include the PI name, the site address, any other locations where the research is done for that site, not everywhere in the study, but for that site. What other locations are they using? Who the IRB is, who the sub-investigators are, what local labs or central labs they're using. Those are the things put on the 1572, and then the protocol title. What is the study? What's the title? What's the number? Who's the sponsor? Those are what's put on there. Now, there's a section on the 1572, like I just said, for sub-investigators, or sub-Is for short. anyone listed in that box, I believe it's box 6 on the 1572, including the principal investigator, have to sign financial disclosure forms. These forms disclose how much financial interest the investigators have in the pharmaceutical sponsor. It doesn't say they're not allowed to have financial interest. It just says if they do, they need to disclose it, and if they don't, they need to put that they don't. So every sponsor has a different threshold for the amount. So I've seen $5,000, I've seen $50,000, I've seen everything in between. Whatever the requirements are for that sponsor, it's clearly listed on that financial disclosure form. And anyone listed in box 6 on this 1572 needs to complete this form. Whether they have financial interest in the company or not, these forms need to get filed, and those go with the sponsor for the essential documents. Now, a final, very important thing. IRB approval. Remember all the stuff we just discussed about how the common rule, or actually even before that, the Belmont report stated that there must be an IRB in charge of studies and all those things. IRBs have to approve sites individually, one by one, whether it's central IRB or local IRB. The IRBs have to approve the sites. They have to give sites formal permission to start screening subjects. They could say no. It doesn't happen that often, but they could say no. There are circumstances where the IRB will not approve a site to screen. Maybe there's some issues with that site, maybe there's some issues with the PI, maybe there's some issues with that protocol the IRB is not comfortable with in the case of a local IRB. The IRB approval, and the way you get IRB approval, the way sites get IRB approval is by completing an IRB approval form. And this form can be found on most IRB websites where they have different portals for sites. There's a handful of commonly used central IRBs, and these forms are found on those websites. You can Google it right now and find it yourself. Just Google Copernicus, Quorum, Sterling, various IRBs. You can Google these things, you can find these forms, and then you can't fill out the forms and submit it if you don't actually have a study, but you can get a sense for what these forms ask. They basically ask who's the PI, who's the coordinator, who are the sub-Is, what are your SOPs when it comes to protecting subjects, who do you plan to recruit, are you recruiting from vulnerable populations, what is the community sentiment in your area in regards to research, what is the staff workload, how many studies is the PI doing, how many studies is the coordinator doing, different things like that. It's about a 13-page form, and this is probably the most time-consuming startup regulatory document for sites, is this IRB approval form. Once the IRB approval form goes through and the site gets IRB approval, the IRB will then send them an informed consent template. This is pretty simple to fill out for the sites. The site puts in, it's already a informed consent template that has been created by the sponsor and approved by the IRB already, but the site needs to put in their specific information. Who's the PI? What's the contact number? What's the address? How much are you paying subjects at your site? Various things like that. What is the 24-hour emergency contact line? Those all go in the template. Then the site sends that template back to the IRB and the sponsor. The sponsor creates a approved informed consent for the site to utilize when they start screening subjects from this. So these are the regulatory startup documents, right? In addition to these things, you also, sponsors and sometimes IRBs even require CVs, medical licenses, IATA certificates, CLIA waiver or CLIA certificate, and the GCP certificate for all staff. All staff. Okay, so that is regulatory startup from site selection visit. So this, and you'll learn this later as you learn the different monitoring visits, this occurs in between site selection visit and right before site initiation visit. This is where regulatory startup factors in and comes into play. So I have a lot of people watching on YouTube that have asked me in the past, hey, what do you do in between site selection visit and site initiation visit? Do you just sit around and do nothing? No. This is what needs to get done. All right? It's time consuming. Obviously, if you have a lot of studies, you're doing a lot of these, right? You should have a system in place. But this is done between, so after SSV and before SIV, which is site selection visit, and then right before site initiation visit, these things get done. As far as regulatory, there's a lot of other stuff too, as far as investigational product, getting the lab supplies, getting the patient-reported outcome diaries, creating the source documents, getting your contract and budget finalized. Those all happen as well, but as far as regulatory, because I wanted to make this part of the video just about regulatory startup. Alright, so this is what happens in regulatory startup. Now, after site initiation visit, which again we'll learn about later, we get to when the study's ongoing. Alright? And when the study's ongoing, we call this regulatory maintenance. One of the most important forms in research is the delegation of duties log, or sometimes called delegation of authorities log. There's sometimes in research, there's like various different names for the same thing, which is why it's confusing for people, understandably so, for people who don't have experience with the research. But just know, anytime you hear delegation and log, no matter what that word in between is, that's what they're talking about. Matter of fact, just when you hear the word delegation and log, you should think of these things. So what the delegation of duties log is it literally, and this is done at SIV, site initiation visit, or prior to it, it lists out everyone who's going to be involved in the study. Literally everybody, not just people from box 6 of the 1572, because that just lists sub-investigators. This lists everybody from PI all the way down to research assistants. Everyone at the site level. Everyone at the site level. So it doesn't include the monitors on there. But it includes everyone at the site level. Who are they? What is their name? What is their signature? Because remember from ALCOA, attributable, we need to be able to see which signature is attributable to what person. When we're looking in a source document and all we see is a signature, we can refer back to the delegation log and see whose signature that is. We can actually see a name associated with that signature, okay? So name, signature, initials, for the same reason as attributable. Then you have start date, so what date did this person start on the study? And then you have an end date. So this is when the study's closed out, or maybe when the staff member is no longer at the site. End date. Alright? And then you have the PI, initial and dating, that they approved this person and they've trained this person to work on the study. There's also another thing on the delegation log which is pretty important. It's all the assessments that they're going to be doing. What duties have they been delegated? Just like the log, the name of the log indicates. What duties have they been delegated? So, informed consent. Who's doing informed consents? Who's doing vital signs? Who's collecting labs? Who's randomizing subjects? It could be more than one person doing different things, but on the bottom of this log is a key with all these assessments, and they have letters or numbers attributed to them. So for example, vital signs could be letter D. Informed consent could be letter A. Letter K might be dispensing study drugs. Letter X might be recruiting patients. Usually there's a lot of duties, and they're all listed out. It's all broken down into all the possible assessments that are done for this particular study. So the schedule of assessments, which we're going to get into in the source document section, just breaks down every assessment for every visit. And then this log will attribute a letter to every staff member that is on this delegation log. What are they doing? So the PI might have every single letter from the key next to their name. The coordinator may have half of them. A sub-I may only have 2. A recruiter might only have 1 letter next to their name. But it doesn't really matter as long as it's there. Who's doing it, who's delegated to do this duty, and their signature, when were they started on the duty, when were they trained for the duty. These are the kind of things that are put on the delegation of duties log. Okay. Another thing very closely related to delegation log, because everything's related, is training log. Let me write this here. This is one form that again is usually done at site initiation visit, and it could be done. This is one of the few regulatory documents that the CRA can actually sign. Okay. So what the training log states is whomever these people are, certainly the ones on box 6 of the 1572, but also anyone on the delegation of duties log, the training log certifies that these people have been trained on the protocol, okay, and on the most recently updated protocol, which we're going to get into right now as well. So the PI, the CRA, the coordinator, anyone can train, any qualified person can train a staff member on the protocol. And let's say the protocol is amended, that training has to be done for everybody on the delegation log as well. So this is done the first time, usually at the site initiation visit, by the CRA. So the CRA will get everybody at that site initiation visit from the site who's there. It's usually not everybody who's on the delegation log, but whomever is available that date. Usually it's the PI and the study coordinator and maybe some of the sub-Is. The CRA trains them on the protocol. Everybody signs the log. Everybody acknowledges they've been trained. And then whomever was not there for the CRA to train, the site is responsible. Typically it's the PI, but it could be the coordinator. Remember, anyone the PI delegates to do this. Any qualified person can train everyone else who missed out. But the point is, everybody's got to fill out this training log. Alright? So that's the training log. Okay, some other things from regulatory maintenance are also things from startup. But let's say the study's going on. It's ongoing. And now there's a new study coordinator. Okay? And the study coordinator also happens to be a sub-investigator, which is often the case. It often happens in these studies. Well, the sub-investigator is going to need to be added to box 6 of the 1572 form. So we're going to need a new version of the 1572 form with the updated investigator, updated sub-I. That same person has to now be added to the delegation of duties log. That same person has to be trained on the training log. That same person has to file a financial disclosure form. So a lot of these things repeat themselves during the regulatory maintenance, but that's one of the things that occurs during regulatory maintenance. Another thing is protocol amendments. This is a big one, okay? So protocols typically get amended between 3 to 4 times per study, per average study. Each time there's a protocol amendment, it requires new training on that new version of the protocol for everybody. Everybody. The PI's responsibility to make sure everyone on the delegation log is trained on the most recently amended protocol. That protocol amendment must be filed. The entire protocol, the new version, must be filed in the regulatory binder. That's part of regulatory maintenance. The protocol signature page for that amendment needs to be signed and filed. The CRA is checking for all this stuff, okay? So that's what happens on amendments. Another thing that amendments trigger are informed consent amendments. If anything has changed in the study, and that's usually what happens in a protocol amendment, something has changed from the previous version, subjects should be notified if it affects them, and most times it does. Even if it's something as simple as we're adding a new ECG procedure at this visit, that could trigger a new informed consent form. And now the site needs to use the newest version of the informed consent. Not only that, But every subject has to reconsent, and we're going to get into this in the site level dynamics later when we get into source documents. But informed consent is sort of like both regulatory and source, because it's written on by sites as a source document. It's put in the patient binder as a source document, but it's also regulatory, as you can see here. So those are the things that are done if a subject does not sign the new amendment, and they're still actively enrolled in the study. And any study assessments have been done since that amendment has come out without the subject signing the informed consent, that is going to be a deviation. And we're going to learn more about what a protocol deviation is, but for now just know any just like the name says, protocol deviation is any deviation from the protocol, whether done intentionally or accidentally. Sometimes they could be done intentionally for matters of patient safety. Other times the site just makes a mistake. They forget to do something. That's a deviation. There's something in regulatory called a deviation log. So a deviation log. And as you can tell, and as you will soon see, there's logs for absolutely everything in research. Just about. But anytime there's a protocol deviation, staff should fill out what the deviation was. And by the way, sometimes the staff is not even aware that there was a deviation, and the CRA has to actually point it out and say, hey, this is a deviation. Fill out the log, and anytime a deviation affects patient safety, the site needs to fill out a deviation form and send it to the IRB. Okay. If it's something that continues to happen, the site is required to do a CAPA plan, corrective action, preventative action. So if it's something that's constantly happening, or if it's something that's important enough and only happened once, but we can't afford to have this happen again, corrective action, preventative action. It's a report that the site creates, sends it to the sponsor for approval, sends it to the IRB, retrains, again, training log. Most likely the PI needs to retrain, or the CRA can retrain the site on whatever it was, right? Same thing with deviation. If there's a deviations, that person should be retrained on the protocol, right? Who made the deviation, they need to be retrained, right? And if it's a CAPA plan, certainly everyone at the site needs to be retrained. Everyone applicable, and anyone whose assessments, who's been delegated that duty. In addition to deviation log, there are adverse event logs, SAE logs, SUSAR reports. So I think now would be a good time to sort of discuss SAEs. Alright, so AEs are part of source documents, and I'm going to talk about AEs in a later section of this video and in the book. For regulatory purposes, when an adverse event is serious as defined by the subject is hospitalized, the subject has really anything that jeopardizes their health, that's an SAE. Okay, serious adverse event. Whenever there's a serious adverse event, the site is required to, within 24 hours of being aware of the SAE, to fill out an initial SAE form, send it to the sponsor and the IRB. That initial SAE form is going to detail everything that the site is aware of. What the SAE is, what happened. Then the PI has to put on there the causality, whether he or she thinks that the IP, the investigational product, had anything to do with the SAE or not. That's the PI's opinion. So causality. And then severity. What's the severity of the SAE? If it was a migraine that required hospitalization, what's the severity? Usually it's on a scale of 1 to 5. Again, only the PI can make those two determinations. right? That's the initial SAE report. This gets sent to the sponsor, this gets sent to the IRB. Once the SAE is resolved, or if a period of time has gone on and it's still not resolved, they send either a final SAE report or a follow-up SAE report. So the site gets more information. By this point, they should have more information on the SAE. The PI gives those two assessments again: causality, severity. And since it's a final SAE report or maybe a follow-up SAE report, they give some information as to what actually occurred since the initial report. So hospital records, maybe medical records from another facility, whatever is applicable, right? They send that to the IRB and sponsor again. The sponsor will look at that based on the PI's assessment of whether it's causal to the IP. The IRB will also look at this, and then they will determine whether it should be a SUSAR, which is a suspected, unexpected, serious adverse reaction. So not every SAE becomes a SUSAR automatically, but if it's suspected to be related to the study drug, and if it's also unexpected, meaning it's something new that they didn't know about before, but usually it's just suspected is enough to trigger a SUSAR, because sponsors tend to be more conservative with these things. Obviously, the FDA is overseeing everything, so it doesn't hurt to be overly cautious. They will send a SUSAR report, which basically is a report that they, along with the IRB, submit to every site participating in this study that's using this investigational product. Even if it's not this study, any site anywhere in the world using this investigational product for any study will receive this SUSAR report. These SUSAR reports need to be signed and acknowledged by those PIs, okay, and then filed in the reg binder. And this is part of regulatory maintenance. So SUSAR reports need to be filed. If a SAE happened here in Costa Mesa, and after my initial and my follow-up and then my final SAE report, the sponsor determines that's a SUSAR, they will send this SUSAR report to every site all across the country. So another site, or even across the world, so another site that's participating in a study with this investigational product, maybe they're in Miami, Florida, they receive a SUSAR report. Hey, this happened at this site, this is what the sponsor believes this is a SUSAR. Please watch out for it at your site. That's essentially what the purpose of these forms are. That PI will sign and acknowledge they received it, file it in the reg binder. Okay, so SUSARs can start piling up really quickly depending on the study, and depending on how many studies, of course, are utilizing the same investigational product, because remember, it could be any study using that IP that gets a SUSAR report. So it doesn't even have to be on your study. It could be another study just using the same investigational product. You'll get a SUSAR report. That's part of regulatory maintenance. It needs to be filed in the reg binder. At your site, whenever there's an SAE, there should be an SAE log, and that should be self-explanatory. That's usually filed in the regulatory binder. Another thing filed in the regulatory binder, another log, is an informed consent log. So remember, every time the informed consent is amended, any actively enrolled subject needs to sign the new amendment, the new informed consent amendment, and the site should document this in the ICF log, which also goes in the regulatory binder. Another part of regulatory maintenance is IRB continuing review. So just like the initial IRB submission, every year there's IRB continuing approval. So the IRB, when they approve a site to do the study, it expires. It's not indefinite. Usually it expires every year, and the site needs to submit for reapproval every year to do the study. Now this is where those deviations are important. If your site is committing a lot of deviations and the IRB determines that it's severely impacting patient safety at your site, they may not reapprove you. They may say, you know what, this site is not going to be reapproved. They may even audit you before they do that, and then make that determination of whether you can, if your site can continue doing that study. So deviations really are important because the IRB has to approve, and this is called continual approval, for the site every year. So that's another regulatory aspect. That's something CRAs should really pay attention to, because if a site is conducting any study activities and this IRB approval lapsed, that's a huge deviation. That could be actually a GCP violation, not just a protocol deviation. Anything that's violating good clinical practice guidelines is a GCP violation. That's a serious offense. So that's regulatory maintenance and startup in a nutshell. We're going to get into, there's a little bit of overlap when we get into source documents, and there's even more overlap when we get into monitoring visits and really breaking down some of the common scenarios CRAs and coordinators deal with on a day-to-day basis. But for now, that's a good overview of regulatory. Okay, so moving right along, we're now going to spend the next hour or so talking about source documents, what they are. We've learned previously in this video, and in the book, it goes into great detail in the book. You've got to get the book if you haven't checked it out already. The Comprehensive Guide to Clinical Research, and also the audiobook, because we go off script for about 8 hours. So we read the book, but then we also go off script on almost every single topic, and it's more like a podcast slash a continuation of the book. So, source documents. First of all, what is the source document, okay? It is any document, data, or record that is the origination of this data. For example, we talked earlier in the video about if somebody takes vital signs on a sticky note as opposed to on the actual source document, that's okay. It needs to be transcribed there, but that source document is not the original source. The sticky note is the original source, and it goes back to ALCOA. So all source documents need to be filled out according to ALCOA standards: attributable, legible, contemporaneous, original, accurate, and complete. Okay? Now, source documents could also be things like lab results. When you send a subject's blood samples to a lab, you'll get lab results in return. That's a source. Source document could be an ECG result. When you screen a subject or any assessment, any visit that has the ECG as an assessment, when you print out that ECG, that's a source. When you get a copy of an ECG report from the ECG central reader, that's a report. That's a source. So source document is really, and it doesn't matter if it's paper or electronic, wherever the data originated from, the first point of entry, that's why it's called the source. The first point of entry, of origination of the data, whether it's paper source, which is still traditional, or as the industry is moving into the next decade, into electronic source, where you can actually enter the original source data on your phone. That's not an EDC. EDC is different. Source data, okay? Source is then entered into the EDC, and CRAs have to verify that data. We're going to get into CRA stuff later point in the video, and in the book we go into it as well. So that's what source is. It's any data that the study requires to be collected, which you can find from the schedule of assessments and the protocol itself, and the origination of this data. That is the source. Medical history, what are the subject's medical records? That's the source. Concomitant medications, which we're going to get into, also known as ConMeds. Where are the pharmacy records? Where are the actual prescription bottles? Where's the prescription? Whatever you have, that's the source. That's the source of the data. Okay, so ALCOA-C is important, and that's the underlying theme of capturing source at the site properly. Has to do with ALCOA principles. All right. Now, informed consent. We're going to go in order, but informed consent we're going to go through some of the common source documents first, and then we're going to give a breakdown of common research site visits for subjects in an average study. Okay, so the first source document is informed consent, and informed consent is also regulatory, okay, because remember, the sponsor has to approve the informed consent, or they have to create it first, and then the IRB has to approve it. And then every time the protocol is amended, it usually triggers an ICF amendment, right? That's all regulatory stuff. We just learned that in a previous section of this video. But ICFs, once patients sign an initial and go through it, and that ICF form goes from being a regulatory document, which is still a regulatory document as far as the template is concerned, but now, once a subject signs it, once the site signs off on it, that's now a subject source, and that should go in the subject source binder. So all these things we're going to talk about go into individual subject source binders if they're paper source. If they're electronic source, which the industry is heading into, they are, whatever the vendor is, they are folders in that platform. Okay? Same thing. A subject source is typically a medical file. You can see them in any office. The traditional medical files with the source documents inside. Okay. Also, the source documents will contain, usually they contain dividers to separate the visits. So this is visit 1, screening, visit 2, randomization, visit 3, visit 4, and so on and so on. They also have dividers for CONMEDs for the subject, medical history for the subject, adverse event log for the subjects, progress notes, everything, lab results, ECG results, all those things, anything that's sourced and pertaining to that subject should go in the subject source binder, right? If the site has only one subject in the study, there should only be one subject source binder. If the site has 10 subjects in the study, the source should be 10 subject source binders. Okay, so that's a source. The first source form that we're going to talk about, which also could be the most important according to the FDA, GCP, and ARB, is the informed consent form, ICFs. So the purpose of the informed consent, as we mentioned earlier, is to provide participants with sufficiently detailed information on the study so they can make an informed, voluntary, and rational decision. This is why we covered the history of GCP, Nuremberg Code, Belmont Report, Declaration of Helsinki, because that's the foundation for why we have informed consents in every study. Okay, and we talked about that previously in the video. If you forgot, just rewind it and go back, right? Or it's also in the book. Alright, so then we talked in the last section on regulatory about amendments. So anytime there's an informed consent amendment, any active subjects in that study need to reconsent on the new informed consent form. If they are no longer in the study, they don't need to come back and sign the new consent form. It's only for actively enrolled subjects. Okay, so that's the informed consent. The informed consent is always done at screening visit. Always. No other assessment that we're going to get into, some of the common assessments for screening visits. No other assessments can be done in the study at all, including collecting a urine analysis to see if they're on drugs, if they're on illicit drugs, including running a breathalyzer test. Nothing can be done prior to the informed consent getting signed by the subject and the staff going through the informed consent and answering all the site questions. There's also a process of consent, right? And we're going to get into what that process of consent is, but most sites usually have a similar type of process of consent in their SOPs. So remember, the informed consent is the first thing. It's the most important thing. Before anything can be done for the study purpose with the subject, site needs to obtain the informed consent. Now, what is the process of consent? The process of consent is how the site obtains consent from subjects. Sponsors may ask this. They ask this at site selection visits. Sites should have this in their SOPs, standard operating procedures, which we're going to get into later on in the video. Right? So, what is a typical process of consent? A typical process of consent goes something like this. Patient arrives to the site. Patient arrives for the screening visit. Patient is put in a quiet room, private room, where they can have sufficient time to go through the informed consent form. There's a delegated staff member there to answer any questions. There's no coercion involved. Alternative treatment options are explained to the subject. All benefits and risks and possible side effects of the study drug are explained. All assessments are understood by the subject. Subject clearly demonstrates an understanding of the study, its purpose, the assessments, what is expected of them. Subject understands that it's voluntary. Their participation is voluntary. Subject understands that they can withdraw at any time. Subject has the right to notify their own primary care physician or other physician that's treating them about their participation in the study. But subject also has the right to keep that information, any study information, confidential. The site needs to respect these patient requests. All of this needs to be documented. The patient is even allowed to take a copy of the consent form home with them prior to making a decision to discuss with their family and friends. Once they sign the consent form and once the PI or the delegated staff member, remember we learned about delegation of duties in the previous section. Once that's done, the subject should be given a copy of their signed consent form. It has the IRB phone number on it. It has the emergency phone number for the site on it. It has all other pertinent information about the study on it. They're given a copy of this informed consent to be taken home with them so they can have a copy of it. This entire process should be documented for every single subject every time there's a new informed consent, anytime a new informed consent is obtained, whether it's at screening or whether it's at a random visit due to an informed consent amendment. This process of consent needs to be documented by the site in the subject source every single time, and preferably in the progress notes of the coordinators or the progress notes of the PIs. This will show that the site is following ALCOA standards. Remember, in research, if it is not documented, there is no proof that it happened. So if the site, let's say the site did all this thing that I mentioned about the process of consent. Let's say the site did it, because they probably did. But nobody bothered to write it down in the source. And then years later, the FDA comes in to audit that study. They look at that subject source, they look at that visit, and they say, okay, I see the informed consent here, but I don't see the process of consent. How do we know what the process of consent was for the site? How do we know the subject was not coerced into signing this? If it's not documented, it didn't happen. This is why it's important to document these things. And as you'll learn in this section of the video with source, documentation is everything. If you don't document, it didn't happen. There's no proof that it happened. So that's informed consent. One final thing about informed consents is the amendments, which I mentioned in regulatory. I'm going to mention again because it's important. And I'm going to mention it again in the monitoring section because it's a very common deviation. Informed consent amendments, anytime an informed consent is amended, any active subject needs to re-sign that new amendment. What often happens at busy sites or at novice sites, or at really any site, it's quite common, is the next time the subject comes in, the site doesn't remember that there was an amendment to the informed consent, or maybe they remember that there was an amendment, but maybe for some reason they're thinking the subject already signed the amendment. Alright, so they don't do it. They don't do it at that visit. And then the CRA comes out for their monitoring visit a couple weeks later, and they find, because they're looking for the informed consent amendment, they know, the CRAs know, that's one of the main things they're looking at when they come in, and they notify the site, hey, this is a deviation, where is this consent? The subject came in, had study procedures done, and they didn't sign the amended informed consent. This is a deviation. Protocol deviation needs to be submitted to the IRB. It needs to be submitted to the sponsor. If it happens routinely on multiple subjects, they might need a CAPA plan, which we discussed earlier. Corrective action, preventative action, right? We got to eliminate these errors at all possible. Technically, the subject not signing that consent form is a safety issue, because what if something changed in the protocol which triggered the ICF amendment, which directly would impact the decision of the subject as to whether they want to continue or not? Maybe there's an assessment, maybe there's a new con med that's not allowed, maybe there's a change to a certain study visit, there's an extra assessment, and the subjects, they don't want to do that. So now they haven't agreed to do that. And so that's deviation, right? So that's important, very important. Make sure you keep a log, and this is why in the regulatory section, in the regulatory binder that we just discussed, there's an informed consent log for every subject. It's a master informed consent log that goes in the regulatory binder. Not only does that track all the informed consent versions that subjects signed, but also the amendments. Okay, so it's not unusual. Remember, on average, a study gets amended, the protocol gets amended 3 to 4 times. It's not unusual for a subject to have to sign 3 or 4 separate informed consents for the same study. And it is also not unusual, unfortunately, for the sites to forget to do these things. And it should be done at the next regularly scheduled subject visit. When informed consent is amended, the subject doesn't have to come in for an unscheduled visit. They don't have to do that. But the subject does have to do this at their next scheduled visit. Right? It's part of good clinical practice. Another important thing for source documents is medical history. Right? We keep talking about regulatory and source. There's a lot of overlap, as you can see, and I'm hoping you're starting to see. That's really, you can break down any clinical trial into 2 aspects, okay? Regulatory and source. That's it. There's nothing else. Everything pertaining to a clinical trial, at least from the site level, and also from the monitor level, but really from the site level, it's either regulatory or it is subject source. That's it. Including investigational product, which is actually both, right? Because there's a master IP accountability log for all the IP that the site has, and then there's individualized subject IP accountability log, which we're going to get into later on in the source. But just wanted to tell you that right now as far as keeping that in mind. It's an easy way to simplify the complex, which is what we're trying to do in this video, in this book, and in the audiobook as well. Okay, so medical history. It's very important for studies for safety of the subjects as well as for the integrity of the data being collected for the study, that a subject medical history is obtained, because it's really the only way to make sure that the subject passes the inclusion-exclusion criteria, which every protocol has. And when we break down the study visits, the various study visits, we're going to explain what an inclusion-exclusion criteria is, because it's extremely important at the screening visit and also at the randomization visit. So, medical history. Oftentimes, the subjects, the study participants, they're not patients of the investigator. They're not patients of the site. They're not known to the site. Somehow they came to the site, they were referred by the site. The site needs to do everything they can to obtain some kind of medical history, to look at the subject's previous medical history conditions. What other conditions do they have besides the indication for the study? Can they confirm the diagnosis that the study is studying? Okay, so if it's an asthma study, can they confirm the diagnosis of asthma somewhere? That's only done for medical history. And then for ConMeds, which is something related to medical history, what other medications is the subject on? Can we confirm? How do we confirm? We confirm from medical records or from pharmacy records. And so in order for the site to obtain these things, they need to have the subjects fill out an authorization to release medical records form. This should be part of the informed consent process. Once they sign the consent form, you move on to the authorization to release medical records forms. Okay? And in some states, like California is one of them, there's a Bill of Rights for experimental subjects. So know the local regulations in your state, in your city, in your county, in your country. Alright, but this typically, the informed consent is universal, and this authorization to release medical records is universal. Okay? Now, what some sites do, because sometimes it's tough to obtain these medical records. You have to rely on other clinics to cooperate. Other clinics are busy. They don't know who you are. You know, it may take time for them to give you the medical records and the pharmacy records, and you may not have time because, as you'll see soon, there's a window between screening and randomization that the protocol allows for, and you typically can't go over that window. Okay, that would be a deviation, and that's not a good way to start a study. So what you would have to do is either get a protocol waiver, which we're going to talk about soon, or you're going to have to screen fail the subject and rescreen them. But you're going to have to ask the sponsor what they prefer if you can't get these records. So what a lot of sites do is they get the patients when they first come in, after they do the consent, after they do the authorization to release records, they give them a face sheet. And on this face sheet, the subject writes their information, date of birth, and then there's questions about medical history. You know these face sheets when you go to a doctor's office as a new patient. They give you a packet to fill out. Well, one of those packets is what's your medical history, what medications are you taking, family history of anything. That's all. The sites do that, and sites should do that in case they can't get these medical records or pharmacy records, which they still should try to get, right? Most sponsors still will require those, but at least they'll have patient self-reported history and medications. At the very minimum, they should have that, and guess what? That's part of source 2. And guess what? If the medical history and the patient face sheet do not correspond and do not add up to the same thing, now you have an issue. The medical history really takes precedence. Unless the subject tells you that the medical history is incorrect, they're actually stopped taking that medication back in 2008. Since then, they've been on this other medication that's not reflected in their medical history or their pharmacy records. So for those things, you got to create note to files, and those things can get complicated. But it's important to know the medical history and the CONMEDs for every subject, because it's the only way to really verify whether they meet the inclusion-exclusion criteria, which we should actually get into right now. The inclusion-exclusion criteria is a list of things that the subject must meet in order to get into the study. That's inclusion criteria, and must not meet to get into the study, which is the exclusion criteria. So these are important, and this is why medical history and CONMEDs are important, because oftentimes there are contraindicated medications with the IP, and the sponsors know this because they wrote the investigator brochure. They know what the pharmacodynamics are of the IP. It's their IP, and in some cases they have an idea that it might be dangerous to mix the IP with this class of drugs, or maybe a certain class of drugs will affect the efficacy data, so they don't want that. They want to exclude those classes of drugs from the study. There's no way for the site to know that unless they have the medical records and the CONMED records, right? So those are two most important things that directly impact patient safety. Is, can you confirm the medications, and can you confirm any other medical conditions that might be exclusionary from the study, and can you also confirm the primary diagnosis? Is it the same thing of what the study calls for? So the inclusion-exclusion criteria is in every protocol. It's extremely important. It should be built into the source documents for visit 1 when you're doing the screening, and visit 2 when you're about to randomize the subject, which we're going to get into as well. But I just wanted to talk to you about the inclusion-exclusion criteria because that directly relates to the medical history and the CONMED logs, or the CONMEDs for the patients, which should be put in CONMED logs in the subject source. Another thing, so we're talking about all these medical records, we're talking about authorizations to release records. Patients' identities while they're in the study and after they're in the study should be de-identified. So the sponsor should never know the name, social security number, or any information about the subject. Their confidentiality is maintained at all times as per HIPAA requirements, and the sponsors, they don't even want to know that, okay? So it's up to the site to de-identify the subjects as best as possible, meaning when they obtain a medical history or a pharmacy record, and there is the patient name, date of birth, social security all over those forms, the site needs to take a Sharpie or something and black out those identifying informations. All you need really is the date of birth and the initials. That's really all you need. Okay, and then the IWRS system is going to assign them a study number at screening, so they're going to get a subject ID, and they're also going to get at randomization, the IWRS will give them a randomization number, right? So that's what the IWRS is for. It gives subjects a subject number, and it randomizes them to a treatment arm. So it gives a randomization number. Okay, so just wanted to tell you that quick little thing about subject de-identification and maintaining subject confidentiality. Okay, now we're going to get into something else that's important, adverse events. So, this is part of SOURCE too. Let's say the subject comes in for a visit and complains of a headache, and the PI writes in the progress notes, as he or she should, because they're maintaining oversight of their study and they're following ALCOA-C principles or guidelines. Subject has a headache. Okay. Now, since the subject told the PI this, when the subject sees the coordinator at the same visit, maybe they don't tell the coordinator that they had a headache. So the coordinator doesn't know that it occurred unless they read the PI progress notes, which, let me break it down for you, they usually don't. Okay? So, and the PI may not tell the coordinator. Maybe it was a minor thing and the PI doesn't think it's important, but he still documented it as an adverse event in the progress note. Well, in every subject source, there is an adverse event log. The same exact thing is in the EDC for that every subject. What are the adverse events? Okay, headache, technically for that visit, should be listed as an adverse event in the log and in the EDC. But right now, under this mock scenario, it's only in the PI's progress notes. So, the visit's complete, subject goes home, CRA comes in, a good CRA comes in, and actually reads the PI notes. And says, okay, headache, alright, let me go to the adverse event log in the subject source binder. Oh, I see that there is no headache listed in the AE log. Let me query it, and this will be an action item. Let me see if it's actually in the EDC. Maybe the site put it in the EDC, just forgot to put it in the source. Nope, I'm in the EDC, it's not in the AE log for this subject. I'm going to query it in here too. That's going to go on my follow-up letter to the site as an action item. Okay? These are things that happen all the time. This is why CRA's job is so important, because if enough AEs are falling through the cracks and not making it to the EDC, that's the data that gets sent to the FDA. And if the FDA approves a drug and there's a whole bunch of AEs missing, and if they actually would have gotten those AEs, maybe they would have made the investigator brochure or the insert package for the possible side effects, right? Like, these things can be missed. And this is why we need good coordinators and good CRAs to catch these things. These are very important. Okay, that's for subject safety. But the second one is the FDA can audit that site and then find all these AEs that are unreported and not put in the EDC. Now it makes the site look bad, it makes the CRA look bad, it makes the sponsor look bad. Maybe now they make the sponsor say, hey, you know, we've been auditing sites and we've found a lot of headaches. We think headache should be listed on the actual medication as a possible side effect. So these are small things that can add up to big things. Okay? When you're dealing with hundreds or thousands of patients in studies, little things add up. And little things can impact the public safety, right? If this drug gets approved, and let's say migraines is a side effect, and somebody gets really bad migraines already as it is, and now they're going to take this drug, they don't see migraines as a side effect because it wasn't reported properly, and now they get a gigantic migraine, okay? And they end up in the hospital. Well, that could have been prevented. And maybe the patient would not have taken that medication if they knew that it causes migraines. So, just another example of how little things add up to big things in clinical research. Now, a special type of adverse event is a serious adverse event, or an SAE. Serious adverse events are not little things like we just said. And even the little things are not little things, as I just illustrated. According to the FDA, an SAE is any adverse drug event occurring at any dose that, in the opinion of either the investigator or sponsor, results in any of the following outcomes. You have death, life-threatening adverse drug experiences, inpatient hospitalization or prolongation of existing hospitalization, persistent or significant incapacity or substantial disruption of the ability to conduct normal life functions, congenital or birth defects, important medical event that may not result in death, be life-threatening, or require hospitalization, but may be considered a serious adverse drug experience when, based upon medical judgment, it may jeopardize the patient or subject, or may require medical or surgical intervention to prevent one of the outcomes listed in this definition. So, that's to the letter the FDA's definition of a SAE. They rarely occur on site, right? Most studies, as we saw when we went through the phases of clinical research, phase 1 through 4, most studies are phase 2 or 3 studies. And those studies are outpatient studies. So patients come into the clinic, they spend a few hours at the clinic, they're given their new IP, they return their old IP, which we're going to get into in the IP reconciliation section of this video, and then they go home. So most of the time that they're in the study, they are not on site. Right? So SAEs are most likely to happen, if they do happen, off-site. And so that means a site won't be made aware of an SAE occurring until after it had occurred. Okay, so let's say a subject is riding his bike, gets hit by a car, and ends up in the ER and gets hospitalized. Technically, that's a SAE. Okay, now it's unlikely related to the drug, but it's still an SAE. So when an SAE occurs, within 24 hours of the site being notified of the SAE, an initial SAE form needs to be completed by the site and sent to the sponsor and the IRB. That's the initial SAE report. Now, what's on this initial SAE report is a brief description of the event, and we discussed this in the regulatory, so I'm just going to go through it because it's important, so I'm just going to go through it again really quickly. A brief description of the event, the severity, and the PI's opinion. Only the PI can put the severity. And the causality. Is it related to the study drug? Only the PI can do that. Okay? And then the outcome, if there is an outcome. Usually there's not. Usually it's ongoing at this point. This is the initial SAE report. Within 24 hours, the site needs to send this to the sponsor and the IRB. When the SAE is over, when the subject has been discharged from the hospital, okay, the SAE is over, the patient's stabilized, that typically ends the SAEs. They will send a final or a follow-up SAE report. This has the same information except now, same information as the initial report, except now they're requesting more of a detailed response. What was the outcome? What was the treatment? What happened? What happened to the study drug? Did the subject have to stop the study drug? Was the subject unblinded? Typically, they will be discharged from the study at this point in most cases because they most likely would not have been continuing to take the investigational product. Okay? Now, breaking the blind. Remember I talked earlier. In the event, let's say in this situation, the ER doctor contacts the site and says, hey, before I begin treatment here at the hospital, I need to know what the subject was taking. I understand they were in a study. We need to know what they're taking. I understand it's double blind, and it may very well be that they were taking placebo, but we don't know. And I don't want to give any medications that may contraindicate with what they've been taking. So then the PI is the only one at the site who can break the blind. So the PI will go to the IRBs system. They will actually have a sealed envelope on site for these situations, and it's usually kept with the investigational product, locked, secured, only the PI can get to it. The PI will break that envelope, they'll get a code, they'll have instructions for how to break the blind. Typically it's to go to the IRBs system, log in, enter the code, break the blind, confirm that they're breaking the blind, and then they'll get the treatment. And then they can tell the ER doctor, hey, this is what the subject was getting. Alright, so that's called breaking the blind. That's a SAE. We talked in the regulatory section about SAEs that become SUSARs and really affect the FDA's decision on whether the drug, that particular SAE, should be a side effect, a known side effect of the drug, and also whether or not the drug is going to get approved or not. Because if they're getting a lot of the same type of SUSARs and the risks do not outweigh the benefits of that particular IP, the FDA is not going to approve that. Another thing I wanted to talk to you about is protocol deviations. Okay, and I know we mentioned it again, and we're going to mention it yet again in the monitoring section, and we mentioned it in the regulatory section of this video. Again, a brief overview because it's related to source. Protocol deviations. Okay, there should be a protocol deviation log in the regulatory binder. Basically, it's any deviation from the protocol, just like the name says. Whether it's on purpose, inadvertently, the site's fault, the subject's fault, the sponsor's fault, whomever's fault it was, doesn't matter, it's a deviation. Technically, the subject cannot continue in the study unless the sponsor allows the subject to continue in the study. If it's something that immediately would impact the safety or efficacy data. If it's something like we talked about earlier, forgetting to sign an amended ICF, that's still a deviation, but they usually don't need protocol waivers for that. The subject can continue in the study. If it's something like missing a consecutive number of days of the investigational product, which we're going to talk about, every protocol has a percentage of compliance in between visits that the subjects must maintain. So they must be relatively compliant with the investigational product. If they've missed too much or not taken enough or taken too much IP outside of the range of the protocol, and we go through this in the book and in the audio book section, the audible section on this off script quite a bit, in those cases you may need a protocol waiver from the sponsor. Alright? So that's what waivers are. You also could get a waiver for inclusion-exclusion criteria. Alright? Let's say the subject meets every criteria except one item. The site could request a waiver to the sponsor. They're unlikely to be granted, but sometimes they are. It just depends. It's a case-by-case basis. And that's who decides that is usually the medical monitor. The medical monitor and the PI of the site. So think of the medical monitor as the PI at the CRO level. They're interacting with PIs at all the sites. Anytime a PI at a site has a particular question about any aspect of the study that requires medical knowledge, they usually talk to the medical monitor, who's typically an MD, physician. Okay? And that's their role. And then they give waivers. So the PI, if the site wants a waiver, the PI typically has to request one from the medical monitor, and then the medical monitor and the PI have discussions, and then they can decide whether they're going to grant the waiver or not. So that's a protocol waiver. In the book we discuss major deviations, minor deviations. I kind of mentioned it here. A minor deviation would be a subject forgetting to sign an informed consent amendment. A major deviation would be randomizing, which we're going to learn what randomizing means soon in this section of the video, I promise. Randomizing a subject that does not belong, which we're going to get into. That would be a major protocol deviation. Okay, now all this source data, and we're going to break down every visit and go through screening visit, randomization visit, visit 3, visit 4, visit 5, however many visits the study has. Source data is being collected based on the schedule of assessments. The schedule of assessments, think of it like the Cliff Notes version of the protocol, okay? What it is, it breaks down every visit for the protocol, and then every visit is broken down into assessments. Which assessments need to be done at that visit? In the book we have an example of what a schedule of assessments looks like. Every visit that a particular study participant goes through, they've got to go through every single assessment on the schedule of assessment. The protocol also has the same exact schedule of assessments in the protocol when they break down every visit, but a quick reference for this would be looking at the schedule of assessments, which is a graph has every visit 1, 2, 3, 4, 5, and beyond. And then it breaks down every assessment in the study, and it has a check mark next to what visit and what assessment gets done at that visit. Okay, so that's the schedule of assessments. All this source data that's collected as per the protocol at every visit gets entered into the electronic data capture system. The EDC is not source. Okay, the EDC is a copy of what is on the subject source. Right? The EDC has to be manually entered by someone at the site and has to be signed off on by the PI at the end of the study. So the source is the source data. That's the original source data, like we discussed. EDC is a transcription of this data in an electronic data capture system. Right? Monitors, we're going to get into, and we already discussed earlier, source data verification and source data review. They're the ones that are making sure that all the information collected on the source throughout the course of the study is consistent with what is entered in the EDC. And if there's some inconsistencies, they issue queries, they issue action items. These are things for the sites to follow up on or clarify. So now we're going to go through a typical course of study visits at a site. And again, protocols are different. Some protocols, this won't apply to every protocol, but as a general rule of thumb, which is what the video and the book are really trying to give an overview on, is the most common type of visit. So the first one is the screening visit. So the screening visit, or in most cases, and again there's exceptions, visit 1. All right, it's the first visit. Before a study participant can be randomized into a study, alright, they must be screened first. And we're going to get into what randomized means in just a little bit. So a screening visit consists of all the assessments on that schedule of assessments we just had on here, that we just talked about. All the assessments that are in the protocol, right, for that visit. Like we said earlier, it starts with informed consent. Nothing else can be done with informed consent. Right? I even went through the typical process of consent for a site. So the next thing typically involves obtaining the medical history, which we talked about, obtaining ConMeds, which we talked about, getting a blood draw for the safety lab assessments, doing an ECG for safety reasons, obtaining vital signs for safety reasons, and then the PI and coordinator, or both, will write their progress notes. They're going to document what happened at the visit. Hopefully they'll document the process of consent, and they will go through as much of the inclusion-exclusion criteria that they can. Remember, they just drew blood for the labs. They sent the labs to a central lab reader. They're not going to have those results during the screening visit. Unless they're using a local lab, sometimes they can get the results the same day. Usually most sites use a central lab, which is a lab that the sponsor has designated to receive all the lab samples, and within usually 3 days the labs will send the results back to the sites by email, by fax. Usually it's one of those two methods. So the PI and the coordinator can go through a lot of the inclusion-exclusion criteria based on medical history, based on ConMeds, if they have those, and self-reported is okay too, based on the other assessments. Usually they can't go through the inclusion-exclusion criteria of stable lab results or non-clinically significant lab results, right? So when the site gets back a lab result or an ECG result, there's going to be abnormalities in many people's labs. The PI needs to designate whether they're clinically significant or not clinically significant. Most inclusion-exclusion criteria, which is the most important thing at screening and randomization visit for the sites to look at, most inclusion-exclusion criteria have an exclusion if there's any clinically significant lab or ECG results. So the ECG results the sites can get instantly at screening visit. The lab results, they've got to wait until the lab vendor sends them the result. Then the PI has to go through it. If everything checks out, if the medical history came in and it confirms what the patient told them, the subject is ready to be randomized at their next visit. Okay. Now, sometimes screening and randomization are the same day. If you can get the lab results on the same day. Usually, there's a window, a period of time that the protocol specifies you have to randomize within 2 weeks of screening, or within 4 weeks, or within 6 weeks of screening, right? So if screening is visit 1, randomization is visit 2, usually. And some protocols are designed differently where you don't actually randomize till visit 3 or 4. But again, for the purposes of just brevity and being clear, and giving a basic overview, visit 1 screening, visit 2 randomization. So the IE criteria has to check out, okay? This could be any amount of time, days, a few days to a few weeks, typically, between screening and randomization. The protocol will specify when. And then the subject should be scheduled to come in if everything on the inclusion-exclusion criteria checks out from the screening visit. Everything. Not one inclusion or exclusion criteria need to be off. All of them. And remember, if the PI feels strongly that the subject is a good fit despite one criteria not being met, they can request a waiver. But they're not granted that often, but they can try. It doesn't hurt to try. Okay, but you have to get that in writing as the site. So if everything checks out during the IE criteria, during the screening visit, during this period of time, the subject's at home during this time. They're not on site this whole time. Most studies are outpatient studies. They'll schedule a randomization visit, or visit 2. And then we're going to get into what happens if the subjects don't meet IE criteria. Okay, we'll get into that. Just so you know now, that's called a screen failure. We're going to cover that briefly. But right now, let's assume the subject qualifies. So they come in to get randomized within the protocol allotted time period. They can't be outside that time period. If the protocol says between 3 days and 3 weeks of screening, you can't bring the subject in the next day after screening. That'd be a deviation. You can't bring the subject 4 weeks after screening. That'd be a deviation. It has to follow the protocol, right? At randomization, right, the site will schedule a randomization. It's also known as a baseline visit. We're going to explain why it's called that. Now, here's why they call visit 2 randomization. The subject will complete, at the randomization visit, will complete all the assessments we mentioned before, from the protocol, from the schedule of assessments. All those assessments. Usually it's labs again, ECGs again. The reason it's called baseline visit is the labs and ECG that will be collected in the vitals and all the safety and efficacy assessments that are collected are the last time those things will be measured prior to the subject receiving study treatment. That's why it's called baseline. Those are the baseline results. That's what's going to be used to measure the progress throughout the study for these same safety and efficacy endpoints. Right? So, at the end of the visit, after all the assessments are done, you don't need to wait for the lab results. We already know the subject's qualified at screening. The labs may come back clinically significant after the subject's randomized, and then it's up to the PI to determine if it's still safe for the subject to continue or whether it's still compliant with the protocol for the subject to continue. Right? But let's say all the assessments are done at randomization. The last thing left to do is to assign them a treatment. That's done through the IWRFs. Now remember, most studies are double-blind and randomized. What double-blind means is the subject won't know what treatment they're getting, and neither will the researchers. The only one that's going to know is the sponsor at the end of the study when they de-identify all the randomization codes. Alright? So, that's double blind. Randomized means it's randomly assigned. The PI doesn't get to pick, the patient doesn't get to choose, nobody gets to choose, the sponsor doesn't even get to choose. They can't cherry pick. The IRBs is random. It's like a slot machine. It's like flipping a coin. Okay? Typically, the treatment options in most studies, and again, I'm just a general overview, they're designed differently. In the book, we discuss different designs of protocols. Just a little bit, because we wanted to still keep it easy to read for a novice. There's either the investigational product, which can also vary at different doses. Right? So various doses of the IP. Placebo, which is an inactive ingredient. It's like taking a water pill. There's nothing in the placebo. Or a current standard of treatment already on the market. But all these things are made to look alike. All these things are made to look similarly. So whoever's dispensing it and the patient won't be able to tell what they're getting. Right? That's what's called blinded or double blinded. The research staff is blinded, the patient is blinded to their treatment. So those are typical treatment arms. These are called treatment arms that the subjects can be randomized to. And again, that is done by the IRBs system. So that's randomization. Now the subject is given the investigational product. See what happens at the IRBs. The IRBs will ask, is this subject ready to be randomized? Yes or no? Site puts yes. They ask to confirm again. Yes. The site will receive a code for that subject. Only the system knows what treatment arm that subject's been assigned to. The site will then go to their IP cabinet and look for that number on the bottle, on the blister pack, whatever it is, and get that IP, get enough of a dose for, and they're already prepackaged, so get enough for their visit, right, to last them until their next visit, which will be visit 3, which could be any time after visit 2. Again, depends on the protocol. It could be a week after visit 2, it could be a month after visit 2, it could be 3 months after visit 2. Typically it's a week or 2 weeks after visit 2 for most phase 2 or 3 studies. Alright, so they're given enough IP to last them until their next visit, the subjects are. They're even given extra because every protocol allows a window in between visits. So, let's say between visit 2 and 3, there's a week. The protocol calls for the subject coming back in a week. Many protocols allow, because they understand patients' schedules may change, sites may not be able to fit in someone on a weekend. Okay? They allow plus or minus 2 days, plus or minus 3 days, plus or minus whatever the protocol states. So they're given enough IP in that bottle, in that blister pack, to last them the allotted window between visits, plus the maximum window outside of the visit. So if they're supposed to take a pill a day and they're supposed to come back in 7 days, but the protocol allows for them to come back up to 10 days later, they'll be given 10 pills. They don't have to take all 10. And the subjects need to be instructed on how to dose properly according to protocol. Just because there's 10 pills in that bottle doesn't mean they need to take all 10 before their next visit. They would be over-compliant in that case. We're going to get into IP accountability a little bit, but a lot in the book. Alright, so that's what happens at a randomization visit. And of course, this is all captured and put in the subject source binder, progress notes, as per ALCOA-C guidelines, and then it's all transcribed into the electronic data capture system. And this repeats every single visit. So then you have a visit 3, and now they're called routine study visits. So visit 3, 4, 5, 6, however many the protocol has, the same process is followed. Assessments are done, safety and efficacy assessments based on the schedule of assessments, based on the protocol. Okay, the IRBs has gone to, you're not randomizing the subject again, but only the IRBs knows what bottle number or blister pack that patient has been assigned to. They're given a randomization code at the randomization visit. And so the IRBs knows what treatment arm the subject is on, and they're going to give the site that corresponding bottle or blister pack number for all their next routine visits that they're going to come into. Now, remember, subjects can quit at any time for any reason, right? There's a special visit for that. It's called early termination visit or completion visit. So, let's say a protocol has a study with 8 visits, okay? You have visit 1 screening, visit 2 randomization, visit 3, 4, 5, 6, 7, and 8, okay? If the subject makes it all the way to visit 8, they just follow this process the whole time. Assessments, IP dispense, IP accountability. What the subject does at each visit, at each routine visit, is they will return either the empty bottle of the IP or the bottle with however many pills are left. The site will do an IP accountability. They're literally going to look at what is 100% compliance, and they're going to look at what the subject's compliance percentage is. Right? They're going to do the math. They're going to actually manually count pills. The CRAs do this too when they're reviewing what the site has been doing with IP accountability. That's called IP accountability. Every protocol allows for a certain variance in compliance. Just like they allow for a variance in the windows, plus or minus 2 days, protocols call for 100% compliance, plus or minus 10, 20, 15, 5, sometimes it's 0, right? But usually they allow for over or under compliance for the IP. If the subject is over that limit or under that limit, it's a deviation, protocol deviation, and then if it's up to the sponsor and the medical monitor to determine if the subject's going to be allowed to continue or not. And again, they're going to need a waiver for that, right? Now, let's say the sponsor decides the subject can't continue. They were over compliant, they took way too many meds, or they were under compliant, they didn't take any of their meds, and the data is no good. The subject needs to come in for an early term visit. At the early term visit, the subject brings back their IP bottles with the leftover pills. The site does all the early term / completion visit assessments, all of them. There's no more IP to dispense. This is the last visit. But there's safety assessments, there's efficacy assessments. They do as much as they can, as much as the subject wants them to do. And then the subject is discharged, right? The subject is given a copy of their records, of their ECG, and if they want, they can come back in and get a copy of their lab results when they come back in. And the subject should be referred back to their primary care physician. If the subject allowed the sites to communicate with their primary care physician. Sometimes the subjects don't allow the sites to communicate with their primary care physician, so the only thing the site can do is give the patient a copy of their lab results, ECG results, and encourage them to go seek treatment. Okay, that's the only thing the site can do if the patient does not want them talking to their primary care physician or other physician about their study participation. Now, let's say the subject completes every visit up to visit 8. They just go through the same thing, and it's the same process, and it's the same exact visit. Okay, it's either called early term, if it's earlier than the protocol calls for, or completion, if it's done according to the protocol. Right, that's the only difference. Now, sometimes subjects can be lost to follow-up. Subjects may not want to be in the study any longer, but they don't feel like talking to the site, and they just cease all communications. Those are called lost to follow-up, LTFU. Right? The site still needs to make attempts to complete this early termination visit and to retrieve the investigational product bottle, because if at one point the subject came in and received IP and now they're lost to follow-up, the site needs to get that back, and they need to do their best to get the patient to come in and do the safety and efficacy assessments one final time. Now, that's not always possible, and it's certainly up to the subject whether they even want to do that or not, but good clinical practice guidelines, mandates, and this has to be documented by the site. Remember ALCOA-C, if you don't document it, it doesn't happen. The site needs to attempt 3 phone calls and a certified letter to the subject, and it all has to be documented. And basically what those calls and letter are saying is, hey, please come back to do an early term visit. It's for your own safety, and please return the investigational product. It's completely voluntary. If you don't want to do any assessments, it's fine. You can just return your IP. We'd really appreciate that. Okay, that has to be documented. 3 calls need to be documented, and a certified letter. The receipt needs to be documented. A copy of the letter needs to be documented. So that's typical study visits. There's also, we didn't really talk about screen fail visits. So if a subject is screened and doesn't meet all the IE criteria, or maybe they do at the screening visit, but then when the site gets the lab results back, the PI says, you know what, this particular result is clinically significant. I don't feel comfortable having this patient in the study. They will be screen failed. Okay. Basically, the site goes into the IWRS system and hits a button for screen fail. Just like you have the option to randomize, which you shouldn't do on accident here because that would screw things up really bad. There's an option to screen fail, and what that does is it officially screen fails the patient. The reason needs to be documented in the subject source. Why did they screen fail? Which inclusion or exclusion criteria did they not meet? And then that gets transferred in the EDC system as well, manually. Okay, so that's screen failure. It's simple. There's another visit we didn't mention. It's called unscheduled visit. And unscheduled visits can be done for any reason. The protocol allows for them most of the time. Usually it's done for a safety issue. Like, let's say at visit 3, the patient came in, did all the assessments at visit 3, and then what happened was the labs came in 3 days later, and the PI, he doesn't think it's significant, but he's not happy with how one of the particular lab results is trending. So, just to be safe, and he's doing his job as a PI, no harm to subjects, remember, patient safety top priority, he'll have the patient come in for an unscheduled visit, meaning it's not part of the protocol, but the patient's going to come in and do a repeat of the labs, right? And also just look at the patient. Maybe there's some other things they could look at. It's up to the PI. This is all usually for medical reasons, safety reasons. So that's an unscheduled visit. Again, it needs to be documented. It doesn't necessarily mean it's an adverse event, because if it's not clinically significant, it's not an adverse event. If it is clinically significant, it is an adverse event. Now, the question is it a serious adverse event? Is it jeopardizing the patient's safety? Whatever the reasons are, the unscheduled visit can be scheduled for the subjects, okay, upon PI's discretion. That pretty much wraps up source documentation, the dynamics at the site level, what typical visits are in a study, how the workflow works at a study. As you can imagine, most of the stuff I talked about is done by coordinators, and then the safety oversight is done by the PIs, and then the monitors review it, and we're going to get into monitoring in the next section of this video. Alright, so now we're going to talk about monitoring visits, and before we get into each individual type of monitoring visit, which I'm about to do, we're going to talk about the process of monitoring, and then I'm going to go through and break down every monitoring visit, what happens, what's typically expected of a CRA at these visits. So monitor is another name for CRA, which is Clinical Research Associate, right? The closest related thing or job to a CRA at a research site, unless you're a large site with for example, universities actually have their own CRAs within the university. If you're at any other site, privately funded industry site, you have coordinators or quality assurance personnel. So these are also what the QA specialist would do at the site level, maybe without the formalities of the follow-up letters, the monitoring reports, and the closeout letter, or sorry, the confirmation letter, the monitoring report, and the follow-up letter. So that is the process of monitoring. It's before any scheduled monitoring visit, the monitor sends out a confirmation letter. So we'll do that first. Confirmation letter. Alright? In this letter, the CRA, first of all, they have a agreed upon date and time that they're going to do the visit at the site. So they will email or call the site and say, hey, does this date work for me to come in for my monitoring visit? The site will either say yes or no, and then they'll go back and forth on a date until they find a date. Once they get that date, the monitor will send a formal confirmation letter to the site. In the confirmation letter, it's going to spell out the date, the time, who's expected to be there. Here's a hint, the PI is always expected to be there, even if they can't. It's documented in the note. And then an agenda of what will take place at this particular monitoring visit. So that's pretty simple, confirmation letter. The next thing, this is where the CRAs really earn their paychecks, and this is what makes monitoring somewhat stressful, in addition to all the SDV, SDR stuff, which we are going to talk about right now as well, okay, is monitoring reports. Alright, these are also called trip reports, annotated trip reports, or just simply monitoring reports. In the monitoring reports, so the confirmation letter is sent out, okay, then the actual monitoring visit occurs. During and immediately after the monitoring visit, the CRA is responsible for completing the monitoring report. This is their deliverable. This is how sponsors and their line managers at the CRO level know that the CRA is doing what they're supposed to do. These are very detailed reports, and in the book we have examples from my own CRO of what one of these monitoring reports actually looks like. But for the purposes of this video, just know that they're extremely detailed, and we're going to get into every single monitoring visit and sort of break that down, and everything we talk about in those upcoming sections of this part of the video go in the monitoring report for that individual visit. So if it's a site selection visit, there's a specific site selection visit monitoring report. If it's a site initiation visit, there's a specific SIV monitoring report, and so on and so on. So that's the monitoring report. This usually takes, the confirmation letter is sent 1 to 2 weeks prior to the actual monitoring visit. The monitoring report is done 1 to 5 days after the monitoring visit. And then there's a follow-up letter. Okay. Now before you get to the follow-up letter, the monitoring report has to be finalized. And what that means is that the sponsor has to approve the monitoring report, and before it even gets to the sponsor, the line manager of the CRA is going to add comments to the monitoring report, ask questions, ask for clarification on certain things, ask for revisions. So it's not uncommon for this monitoring report to go through a couple of rounds of revisions between the CRA and their line manager until they agree that it's okay now to send it to the sponsor. So this goes to the line manager. Every organization has a different name for this person. It could be lead CRA, it could be project manager. Typically it's your boss, your direct report, who you directly report to as a CRA. It's one of these people, right? You go through some revisions. This whole process takes another week or so. Once it's ready to be sent to once the line manager is comfortable sending it to the sponsor, they send it to the sponsor for review, okay? And the sponsor is required by the 2016 GCP revisions to have oversight of their study. So back in the day, the sponsor could outsource all these things to the CRO and the CRAs and say, hey, we're not responsible, we outsource this to the CRO. Now, the FDA mandates that they actually have oversight and they are held responsible for what goes on in the study. So they review every single monitoring report, right? And sometimes they have questions. So sometimes they require revisions. So they'll tell the line manager, and the line manager will tell the CRA to revise it. This could take another week, all right? And this whole time, now we're talking about a couple weeks, this monitor has already done probably another 3 monitoring visits. So this whole process repeats with their other visits while this report is getting finalized. So things can add up really quickly, things can get stressful really quickly, and this is important for everyone to understand. These are the things that make CRA stressed out: the details, the staying on top of things, the getting your report submitted in a timely manner, revising based on sponsor questions, line manager questions, while maintaining your current workload. It starts piling up very quickly. All right, once the report is finalized, okay, this is a finished product. Now the CRA can use this finalized report to send the site the follow-up letter. All right. So from these 3 things, the site only receives the confirmation letter and the follow-up letter. The site does not receive the monitoring report. That's for the sponsor. Once the report is finalized, it goes in the trial master file system, which we discussed in the regulatory section. Trial master file. These days they're electronic, so these are called TMFs or eTMFs for electronic. So all these things go in the trial master file. All right, confirmation letter, finalized monitoring report, and then the follow-up letter. And on the follow-up letter, the purpose for the follow-up letter, because remember, the site is not getting the actual monitoring report. So how does the site know what to do as far as their action items? How does the site know what action items they need to complete? Well, that's all spelled out in the follow-up letter, all right? The action items. So this is also sent to the site. The confirmation letter, the follow-up letter sent to the site. They're also all sent to the line manager. They're also all uploaded into the trial master file system, which, remember, is like a master regulatory binder for every site. So they're separated in different folders based on site for that study. They have all the essential documents, SUSAR reports, everything. In addition to that, they have all the internal reports that are generated by the CRAs for the purposes of the study. And the sponsor, this can be audited by the FDA at any time, and the sponsor can maintain oversight by having access to this trial master file. So that's the process for going through a typical monitoring visit. Any visit, we're about to break down all the visits right now, starting with site selection visit, starting with SIV, then going into interim monitoring visit, closeout visit. There's also a few other visits that we're going to briefly mention, but this is, in a nutshell, the process for scheduling, completing, and then following up with the site for any monitoring visit. Right now we're going to get into, before we break down every monitoring visit, we're going to get into a breakdown of what the difference is between SDV and SDR, the 2 main functions of any monitor. So we're going to do that right now. Alright, so now we're going to get into SDV and SDR, the 2 main functions when it comes to source documents for a CRA. Alright, so there's 2, but there's also another 2. There's regulatory, and then there's investigational product accountability. So monitors do all these things, alright? They review source, they do regulatory, and they review, and when I say do regulatory, I mean they review it. And then they review IP, which is investigational product accountability, right? So we We're going to talk about these two when we get into breaking down each individual monitoring visit, but for right now, before we even get to that, I just wanted you, the viewer, to have an overview of the source when it comes to the source, because that's a huge component. I mean, this is data. These things are not data for the study. Well, the IP accountability kind of is, but it's really, the IP accountability is integrated into source. But it's so important it's treated separately, and we're going to get into that when we break down each monitoring visit. But source, this is the end product of the study, is data. That's all clinical trials are after. Regulatory is a necessary evil. Nobody cares about that. They care about the data. Okay, so this is SDV, which is source data verification, and SDR, which is source data review, are the two functions when it comes to source that monitors are responsible for. So source data verification is the easy one, okay? You look at what's on the source. Every time you go to a site and there's been new subject data from the site on the source, it's recorded on the source, and then it's also put into the EDC system. So all the monitors are doing is looking at the source and then making sure that it's correlated to what's in the EDC. If there's any discrepancies, if there's anything missing, if there's something that's unclear, monitors issue a query in the EDC, and they also issue an action item, like we mentioned earlier, on their follow-up letters. Okay, so that's SDV. If that was all a monitor did, there would be no demand for monitors. There would be no educational requirements for monitors. It's not that difficult to do SDV. It's just you're looking at what's on a piece of paper, or it could be eSource too, but for the purposes of clarity, you're looking at a piece of paper, you're logging into an EDC system, and you're making sure that the data matches and correlates. It's not that difficult, alright? SDR, source data review. Okay, that's where the CRAs earn their paycheck. Okay, and this earn paycheck, it's really from this, and this is what goes into those monitoring reports, the details. What source data review is it's understanding what that data actually is, understanding how it applies to the protocol. Is it a protocol deviation? Is the data, do the subjects actually qualify for the study with the inclusion-exclusion criteria? That's source data review. Source data verification is just looking at what's in the source and correlating it with what's in the EDC system. Source data review is looking what's in the source, understanding it, but then making sure that it also is compliant with the protocol. Because remember, we talked about it in source documents. Protocol deviations occur frequently. Protocol deviations occur all the time. Inclusion-exclusion criteria those are the criteria that determine whether a subject qualifies for a study and gets randomized or not. Alright? If a site is inadvertently committing protocol deviations, or if a site is inadvertently not following the inclusion-exclusion criteria, then it all boils down to the CRA to find those things. And that's what source data review is. Is the data compliant with the protocol? Is the data compliant with GCP? ICH, GCP, which we discussed, and the protocol. So this SDR is the real reason why there are almost mandatory previous clinical research experience for CRAs. Right? Before anybody hires a CRA, they want to make sure that they have some kind of working knowledge of research because of this. Not because of SDV, but because of SDR. Understanding how the data corresponds to ICH GCP principles, and then understanding how it impacts the protocol. Is it compliant with the protocol? Is it not? There's also safety issues with source data review. For example, CONMEDs, okay, which we discussed earlier, or medical history. Okay, sometimes these are written down, but they actually are excluded from the study. But the way they're written down, the protocol maybe only listed the class of drugs that are excluded, but the site just put the individual drug. Alright, so The site maybe thinks they're compliant with the protocol because they don't see that individual drug name in the protocol, but they see the class of drugs in there, but they don't understand that that's the same thing. So you need a CRA who understands these things. This is also why therapeutic indication experience, in addition to clinical research experience, is important for CRAs. Same thing with medical history, okay? Sometimes medical histories directly impact inclusion-exclusion criteria, and the CRA really has to read between the lines, read through the progress notes. Another thing is adverse events, okay? Sometimes adverse events are written down in the progress notes, but the coordinator doesn't see it, so it really means the CRA has to pay attention. This is all source data review, all of it. What's on the source? Is it compliant with the protocol? Is it compliant with good clinical practice? Obviously, you need a deep understanding of good clinical practice, you need a deep understanding of the protocol, and you need a solid understanding of the therapeutic indication, including the ConMeds, the medical history, and AEs, and read between the lines for all these things. So now we're going to break down every single kind of monitoring visit, the most common ones, and then a few of the lesser common ones in the next part of this video. So now we're going to break down the most common types of monitoring visits, and at the end we'll leave a little bit of time for some of the lesser known monitoring visits. So I just explained the process, I just explained what SDV and SDR are. We've gone through, if you've been watching this video this entire time, first of all, thank you, congratulations, you know all the background information and context. Here in the book, it's just in depth, and in the audio book, we just go off script, and you're going to get a lot more context, and you need that if you're trying to get into this industry, or if you're already in it, but you're just trying to master your skills. So right now we're going to break down every single monitoring visit. We'll start with the site selection visit, also known as SSV. Sometimes it's known as a PSSV. SQV for site qualification visit. So SSV, site selection visit. This is where the CRAs are sent out to the sites to see if they qualify to participate in the study as a research site. So some of the things they look at are site capabilities, site experience, PI experience, study coordinator experience, who their IRB is. Can they use a central IRB? Do they have to use a local IRB? What is their lab situation? Can they use the central lab? Do they have to use their own local lab? What other competing trials are there? And most importantly, patient recruitment capabilities. Where are these patients coming from for this particular study? What kind of database do you have? Is it the PI's database? Is it the site's database? What's the electronic medical records or medical records? How do you use the records? How do you get the records? Is it paper-based records? Is it electronic records? Same thing with source. A lot of sites are now using eSource, my own included. eSource, paper source. The CRA is getting all this information in regards to the site to send the report. Remember, we just talked about the monitoring reports, to send a detailed report to the sponsor about the site's capabilities. And then ultimately it's the sponsor's decision to select the site or not. But the CRA has tremendous influence over this aspect because the CRA is the one that actually visited the site, toured the site, understands the site's capabilities, understands the experience of the site. Another thing they look at is the process of informed consent. So the CRA might ask you for your SOPs. They might ask the site for the SOPs. The site does not have to give them the entire SOPs. Just the process of consent is good enough. Maybe there's certain nuances with the protocol that requires an additional SOP to either be created by the site or perhaps it's an unblinded study, so you need one unblinded staff member whereas everyone else is blinded. You have one unblinded staff member that's in charge of IP administration, but everyone else is blinded. So you might need a separate SOP for this. You might need a separate personnel for this. So they ascertain all these things. They get the good clinical practice certificates, they get the CVs of all the staff that are going to be involved, and then they conduct a tour of the site. So that's a site selection visit in a nutshell. Now you got the site initiation visit, okay? And sometimes the CRA that does the site selection visit is not the same CRA that's doing the SIV, alright? Usually, at some CROs, again, sometimes it is the same person across the board, but usually at some of these bigger CROs, the CRA that does site selection visit only does site selection visits, okay? And then the site, once the site is selected for the study, they're assigned their own monitor. Again, sometimes it's the same person, sometimes it's not. They're assigned their own monitor, and hopefully they keep the same monitor throughout the duration of the clinical trial. But as you know, or maybe you don't know, but you will soon know if you buy the audiobook and you read the actual book, CRA turnover, right? It's it's a well-known fact in the industry that the industry has a problem retaining CRAs. So it's not uncommon to get multiple CRAs throughout the course of the study for a site. But anyways, let's get into site initiation visits. So once the site is selected for the study, the site needs to get activated. Okay, and what does that mean? Well, you got to make sure in between site selection and site initiation, remember we discussed earlier, it's startup regulatory. Startup regulatory. There's also contracts and budgets that the site needs to get completed. And part of the startup regulatory includes IRB approval. Okay? And ICF and protocol approval. All these things occur before SIV, right? Furthermore, the site should have their investigational product on hand prior to screening any patients. Lab kits, EDC access, IWRS access, right? So all these things, the startup regulatory documents need to be complete prior to the SIV, the contracts and budgets need to be complete prior to the SIV, site IRB approval needs to be complete prior to the SIV, informed consent approval prior to the SIV, the site should have their investigational product prior to the SIV, lab kits, EDC access, IWRS access. That's all prior to the SIV. Okay? If these things are there, the CRA will come out to do a site initiation visit. Again, they follow the same process. Confirmation letter, then the monitoring report, then the follow-up letter. At the SIV, probably the most important thing they do is protocol training. So the sponsor provides the slides, protocol slides, for the CRA to review, hopefully with the PI, with the study coordinator, and with the other relevant staff members, anyone who's on that 1572, right? So once the protocol training is done, the CRA will fill out the site training log, alright? They're going to sign that they're the trainer. This is one of the only forms, one of the only regulatory forms at the site that the CRA can fill out besides the monitoring visit log, which is just, it's pretty much a sign-in sheet for the CRAs each time they go to the site. But the training log, it's in the reg binder. Remember we discussed the regulatory documents, regulatory binder. Training log's important. This is probably the first time the site's going to be trained on the protocol. Alright, so that needs to be documented in the training log. Another thing that needs to be completed at the SIV is the delegation of duties log, or delegation of authorities log. It has many different names. Those are the two most common ones. Alright, this one the CRA cannot complete, but the CRA needs to make sure that the PI is listing everybody who's going to be involved in the study for any duties. This includes recruiters, this includes contract and budget people, this includes regulatory people, this includes all the sub-Is, coordinators, research assistants, phlebotomists, everyone who's involved in the study goes on the delegation log. The PI will put a start date, obviously their name, the duties they're doing, the start date, their end date, which will be blank for now. So the start date, everyone should have a start date of the date of the SIV on there. Alright, and then the PI will initial Sometimes they'll initial and date the start date and the end date. Sometimes it's just the end date. So every form is a little bit different. But this needs to be obtained by the CRA at the SIV. You've got to make sure, if you're the CRA, that you actually do this. Okay? No one else is going to do the training for you. Any staff that are not there for this particular training need to be trained on the protocol by somebody at the staff. If they don't, technically they can't do any of these things for the study. People have to be trained on the most recent version of the protocol prior to doing anything on the study. So you got to collect this as a CRA. You got to do this and collect it. You got to collect a copy of this to attach it to your report. You got to get an approval, IRB approval letter. Get a copy of that if the sponsor doesn't already have that. Get a copy of the startup regulatory. The only thing you don't need a copy of is the contract and budget, but you do need to confirm with somebody at the site level that they have an executed contract and budget in place. Now, if everything is here that I mentioned and everything's done, the site can technically start screening subjects the very next day or even that day after the training. If any of these things are missing, alright, that might delay the site being able to screen subjects. For example, if the site hasn't received their investigational product, they probably can't screen subjects, right? Unless it's a study where IP is delivered on time, so they can screen, and then that usually triggers the IP to be shipped to the site prior to randomization. Because remember, you don't dispense IP at screening, you only do it at randomization. But some studies, you have to follow the actual monitoring plan for that. Now lab kits, if the site doesn't have screening lab kits, they probably can't screen any subjects. You've got to make sure that's there. EDC access. At least the coordinator and the PI need to have access to the EDC. If they don't, and this often requires training for the sites, they don't just give you EDC access. The coordinators, the PIs, any other staff members that are going to get EDC access have to receive the training. IWRS access, same thing. Okay? They need these things before they can start screening. You can't screen a subject without having access to the IWRS system. So all these things are being done at the SIV. You're making sure all this stuff is occurring. You're putting as much details as you can in your report, and then any action items that are preventing the sites from beginning enrollment and beginning screening need to be put in there. Need to be made clear to the site once the sponsor approves the report. Needs to be made clear to the site in the follow-up letter. Okay? Next visit. Interim monitoring visit. Usually it's done within 2 weeks after randomizing the first subject. So, let's say a site has been activated, they screen somebody, and then they randomize that subject as per protocol. Within 2 weeks of that randomization, the CRA should come out and do their interim monitoring visit. Right? Now, in this visit, you're going to do all the SDV, SDR stuff we discussed as the CRA. You're going to look at regulatory, right? Any new staff members added to delegation log, any new protocol amendments, any new informed consents, any SAEs occurred. This is part of SDR, SAE, AE, okay? Regulatory, new staff, trainings, anything like that needs to be done at the IMVs. And then investigation of product, accountability. Alright? A common deviation is subjects that are not being compliant with the IP for the protocol, and maybe the site is unaware. That's the CRA's job. Right? So this is what's going on at the IMVs. And these go on every 6 to 8 weeks. So let's say you came out 2 weeks after randomizing. Usually, and again, every monitoring plan is different. Usually, and in the book we go in depth on some of the monitoring plans and why they're there, but for the purposes of this video, it's just, that would be too much information. Every 6 to 8 weeks, the monitor will come back for another IMV. And those are going to be called, if this first one was called IMV1, the next one's going to be called IMV2, the next one IMV3, and so on, okay, until the last subject data has been captured, and then you have a closeout visit. Now, it's important to know, during the course of the study, there are periodic locks to the EDC. These are called database locks, right? These are when all the data for that particular time, any data up until the date of the database lock needs to be fully SDV'd and SDR'd. Based on the monitoring plan, it doesn't mean 100% SDV, although most of the times it does, right? And I guess that's another thing we can discuss right now is traditional monitoring, which requires 100% SDV and SDR. So there's traditional monitoring. This is still the majority of trials, although we're starting to move towards a risk-based system. So traditional monitoring, 100% SDV and SDR. Risk-based monitoring, which is a new strategy that sponsors are starting to be more comfortable with, especially after the 2016 GCP revisions, which put the emphasis on the responsibility of the oversight of the study to the sponsor. Okay, so risk-based monitoring means every site is going to be monitored a little differently based on their risk profile. So you may not need 100% SDV, SDR for every site. You may only need it for some sites. Okay, other sites might only need 25% SDV, and the monitoring plan and the site's risk profile is going to let the CRA know what to monitor, what that 25% should be focused on. All right, that's about as much as you should know. When you're assigned to a study, they're going to let you know this in detail. They're going to train you on that. But that's the two monitoring strategies: traditional and risk-based. Sometimes there's remote monitoring, which is actually a tactic or a strategy used by sponsors that could be for either traditional or risk-based. So instead of actually coming to the site, instead of the CRA actually coming to the site, under remote monitoring, again, whether it's traditional monitoring or risk-based monitoring, rather than coming to the site, they can just do it remotely. The site will upload their source documents somewhere, and then the CRA will look at the SDV in the EDC that way. So that's remote monitoring. So those are database locks. They occur regularly, every quarter, usually quarterly database locks. The purpose is for the sponsor to look at the data, analyze it, see if they're liking the safety and efficacy. They can end the study if they don't like what they're seeing as far as the safety and efficacy. So that's important, and that's when the monitors and the sites are pressured for getting queries answered and things like that. Okay. So finally, when the last subject visit, last subject in the EDC has been reviewed and locked, okay, there's a closeout visit. The closeout visit is essentially the inverse of the site initiation visit. So remember this delegation of duties log at the SIV, when everybody had a start date? Well, now at the closeout visit, everyone's going to have an end date. Okay, and some people may already have end dates because they left the site, but for everyone else, the end date will be the date of closeout visit. Just like how in the SIV you needed IRB approval, now you need IRB closure at the site. So you need proof. You need to notify the IRB, and this is the site, but the CRA is making sure that this occurs. The CRA is sending or making sure that the coordinator is sending the IRB confirmation that the study is closed. There's also archiving records. Archiving. So this is how long the site needs to keep the study records, either on site or outsourced to a place like Iron Mountain. Whatever the case may be, whatever the site's SOPs might be, the CRA needs to put this in their report. They need to make sure it's occurring, And some studies require the site to keep records for 5 years, some 10, some 15. I've seen as long as 45 years that the site is supposed to maintain records of the archives. So IRB closure, IP, final reconciliation. All the IP used and unused in the site's locked cabinet or locked drug storage room needs to be accounted for in the master IP accountability log, and it has to match up with the subject accountability log. And it either needs to be destroyed on site, or the CRA needs to physically ship it back to the sponsor based on sponsor or CRO requirements. They'll let you know. The sponsor and the CRO will let you know which of these two. But the IP is either destroyed on site by the site or sent back to the sponsor or a vendor based on the sponsor or CRO requirements. Right? There's two other. These are the most common monitoring visits. There's two other mini, less common monitoring visits. One is called a booster visit. Booster visit, it's all about recruitment. Okay, this is usually when they send out a CRA. Sometimes it's the same CRA, sometimes it's a special person. They're in charge of making sure that the site is screening. And maybe they try to find out why the site is not screening like they said they would. They send out somebody to do a booster visit when the site is not meeting their enrollment projections. And that's something, by the way, that monitors are looking at and commenting in their reports at every IMV. Is the site recruiting as scheduled? Are they on track to meet their enrollment expectations? If not, and if that doesn't change for a few monitoring visits, they send out someone to do a booster visit. Sometimes it's the CRA themselves. Hey, why are you not, what's going on? Maybe we can help. Maybe we can share best practices from other sites that are doing well at recruiting. That's a booster visit. Same process, okay? Report, confirmation letter, report, follow-up letter. There's also pre-audit visits. These are even less frequent than booster visits. Sometimes the sponsor or the FDA or the IRB has notified the site that they will audit them. Well, if there's time, the CRA might be sent out to do a pre-audit visit, where they're essentially doing everything that you would do at an IMV, but you're just trying to clean up things before the auditors come in. So that's the most common types of monitoring visits, with some less common ones, and in a nutshell. So hopefully that helps. So we're going to wrap up this mega video. Thank you very much for watching. By the way, make sure if you liked it, you really have to buy the book. You don't have to, but I would very much appreciate it. But you should. If you are looking to get into this career, it's a must. If you're looking to expand your career, it's a must if you need an overview of some of these concepts, any of these concepts. The problem with our industry is it's very specialized, so people either become specialized something at the site level, something at the CRO level, or something at the sponsor level, or maybe one of the vendors in the industry, but very few generalists out there. And I really think the best defense and the best thing you can do for your career is to be a generalist. So, it just took me until recently, until I started learning more about CRO and sponsor dynamics, because I was at the site level for most of my career. So you can always expand. And then the audiobook on Audible, the Comprehensive Guide to Clinical Research, you need to get it because we go off script. Chris and I, who are the authors of this book, we read it, but then we pause and go off script on many of the different topics. In fact, every single section we go off script, talk about real life examples we've had, funny stories. We try to make it entertaining. So get the audiobook, get the book. Wanted to wrap up with site, CRO, and sponsor dynamics. So, and then this is also career trajectory for everybody, okay? So, whether you're in the industry, or you want to be, or you want to do something else, if you're trying to improve your career prospects in this industry, this might be important because, like I said, there's a lot of specialists. All of these require specialization, but if you want to forever be in demand regardless of what the market says. Be a generalist, if at all possible. So from the site level, during the course of this video, we've already discussed many of the positions, but it's PI. This typically requires an MD or a DO, right? There's sub-Is. These could be MDs, DOs, also nurse practitioners, physician assistants, RNs, and even coordinators. But coordinators are its own category. So study coordinators or clinical research coordinators. These are really the backbone of the clinical trial. They do all the work at the site level. Then there's people underneath the coordinators as well. Research assistants. All right, that's a great entry-level position. These could be MAs, medical assistants. They could be just somebody fresh out of college. They could be literally anything, okay? Research assistant's a great place to get started. You're going to probably, if you're an MA, you're probably going to start out drawing blood, doing vital signs, doing ECGs. You're, of course, going to get trained on everything, all ICH, GCP. That's the beautiful thing about this industry is when you get started, you get trained on everything that, whether you're an MA or a PI, you get trained on everything the PI, maybe even more, because you're probably going to be doing IATA, like we discussed in the beginning. But GCP, ICH, all that stuff, you're going to have experience on your CV, and you can start climbing up. Obviously, you probably will never be able to be a PI, but climbing up to coordinator, maybe sub-I, and then climbing horizontally to the CRO, which we're going to get into right now. But one more thing I wanted to add, we briefly mentioned it in the video, we discuss it quite a bit in the book and in the audiobook. Patient recruiter. A lot of people, okay, studies, as we've discussed, most of them are behind on enrollment. Every site could use recruitment help. If you want to get at the site level, which in my opinion is the best entry level into the industry, because then you could work your way up and out to CRO and sponsor, is start with patient recruitment. Get good at recruiting, right? Usually involves going out, doing lunch and learns with doctors, lunch and learns with the community, generating business for the site in terms of patient referrals, in terms of maybe physician referrals from colleagues of your PI perhaps in the industry. These people don't have time. PIs don't have time, sub-Is, coordinators, even research assistants don't have time. If you have the time, you can go refer or get referrals from PI colleagues. You can go build and establish relationships for sites. So it's patient recruitment and it's just general biz dev. Maybe you can get studies, help the site get studies. You go on clinicaltrials.gov for that. You can help the sites establish new relationships with community resources in your neighborhood, other physicians. Generate business, patients, physicians, studies. These are what the sites live on. Okay. Then you can climb your way up the ladder and horizontally. So a lot of people tell me, hey, I want to be a CRA, which is going to be over here, but I have no experience. Well, based on your educational levels, you got to figure out where you could start. All right? I think patient recruitment is the most underrated, least talked about entry-level position in the industry. Obviously, if you can get to be a research assistant or an entry-level coordinator, go for it. CROs, okay, at the CRO and sponsor level, there are the CRAs, which we discussed quite a bit. Typically, they will never hire a CRA without any experience, except if they're a BS, Bachelor of Science, RN, registered nurse. I've seen many RN person become a CRA with zero experience. Even more so if you have experience as a coordinator and you are an RN, you're almost a shoo-in to be a CRA. Okay, a position underneath the CRAs is the in-house CRA. There's also trial master file specialists. There's also clinical trial assistants at the CRO and sponsor level. And there's PMs, project managers. Okay, these are probably comparable positions, although CRAs actually make more. Okay, so oftentimes in-house CRAs, they will hire for those positions with no research experience, and then what you're doing is you're helping manage the trial master file system, which we've discussed earlier. You're helping sites with study startup and site activation. You're the one emailing the sites or calling the sites when they're at the site selection visit. In between site selection visit and site initiation visit, you're the one calling the site, following up with the site saying, hey, where's the updated financial disclosure form? How far away are you from IRB approval? You're the one helping, assisting the CRA doing these things, because the CRA is busy. They're out doing other monitoring visits. So these are these specialist positions here, in-house CRA, trial master file specialist. That's when you obtain the essential documents, you organize the TMF based on your CRO's naming conventions, clinical trial assistant, clinical trial administrator. If you go on Indeed, if you go on ZipRecruiter and just type in clinical research, you're going to see dozens of these positions. In the book, we're going to try to give you examples of more with salaries. Right? At the sponsor level, you have biostats. Another thing here is medical monitor for the CRO or the sponsor. Think of the medical monitor as the PI for the study at the sponsor or CRO level. They're the ones that we discuss in this video interacting with the PIs when there's protocol waivers to be given. These are always going to be MDs or DOs. Alright, so these are some of the positions. There's tons of other ones. Alright, regulatory affairs, which we briefly discussed here. These are the sponsor, CRO, and site dynamics. And then vendor, tons of vendors. There's like 11 vendors per study. So there's a lot of places you can get involved and get started in this industry. And then there's a lot of places you can go, including maybe you want to own a site. Maybe you want to own a CRO. There's tons of things. You could be and by the way, all these positions, you can be a contractor as well. Once you get experience, you can be a consultant. There's a lot of places you can get into, but getting in is the most important thing. So just wanted to share a little bit of the site, CRO, and sponsor inner workings and dynamics with you all. Thank you again for watching this video. Make sure that you check out the book, the audio book, and hopefully it's able to help you in your career. Take care.